Imagine discovering an entirely new kind of human. Not from a skull in a museum, not from cave paintings or ancient campfires, not from any of the usual things we associate with uncovering the past. Just from a tiny fragment of bone, smaller than your pinky fingernail, and the DNA locked inside it. No face, no name, no story told around any fire we know of. Just a genetic signature whispering across 50,000 years, saying we were here. That's the Denisovans, and the more scientists have learned about them, the more it becomes clear that almost everything we assumed at the start was wrong. For decades, the human family tree looked relatively tidy. Modern humans evolved in Africa, spread outward, encountered Neanderthals in Europe and Western Asia, interbred a little, and then Neanderthals disappeared. That was the broad story most of us were taught, clean enough to fit on a diagram. But then a single finger bone found in a Siberian cave pulled the whole diagram apart. There was a third group, a group that ranged not just across Siberia, but possibly across a territory stretching from the frozen north all the way into the tropics. A group genetically distinct enough to be their own lineage, yet close enough to modern humans that when we met, we had children together. A group whose DNA still lives inside millions of people today, quietly shaping immune systems, altitude tolerance, and biology in ways we're only beginning to understand. So who were the Denisovans really? Not the simplified version, the cautious, evidence-based, genuinely strange and wonderful version. That's what this is about tonight. Before we go any further, a quick thing. If you're listening right now, wherever you are, whatever time it is, drop it in the comments. Seriously, I read them. It's one of my favourite things. Berlin at breakfast. Manila just before midnight. Someone in New Zealand who swears this helps them sleep better than anything else they've tried. The fact that this reaches people across every time zone simultaneously still gets me. So tell me where you are, and if you want to listen without any ads and without music underneath the narration, the cleanest version of this is on Spotify. Following the channel there is honestly one of the best ways to support what I make. The link is in the description. It costs nothing, it just helps. Now, back to the mystery. What's unusual about the Denisovan story isn't just that we found a new human group. It's how we found them, and what that method of discovery forces us to admit about everything we think we know. Normally, paleoanthropology works like this. You find bones. You study the shape of a skull, the thickness of a brow ridge, the proportions of a jaw. You compare those features to other fossils, argue about them for decades, and eventually reach a rough consensus. It's painstaking, contentious, and deeply human work. The Denisovans skipped almost all of that. We identified them genetically before we had any meaningful picture of what they looked like. We knew their relationship to Neanderthals before we knew their face. We knew which modern populations carry their DNA before we could say with confidence where most of them actually live. In the history of science, this is genuinely unprecedented. An entire branch of the human family recognized not by its bones, but by its code. And that's where the misunderstandings begin to pile up. Because when a discovery comes through DNA first, the story that forms in the public imagination tends to be either too simple or too dramatic. Denisovans get described as a shadow species, a ghost lineage, something alien and separate from us. Or they get absorbed into a tidy narrative where ancient humans mingled freely and everything connects neatly. Neither version is quite right. The real picture is messier, more uncertain, and honestly more interesting than either of those framings. What the evidence actually shows is a group of humans, or rather several distinct populations that we're grouping under one name, who lived across an enormous range, adapted to wildly different environments, interbred with both Neanderthals and modern humans at different times in different places, and then disappeared in ways we still don't fully understand. They were not one thing. They were not a footnote. And in a very real biological sense, for some people on Earth today, they never fully went away. The science here is careful work. Where findings are well supported, we'll say so. Where something is a working hypothesis, where researchers disagree, where the data is genuinely incomplete, we'll say that too. Part of what makes this story worth sitting with is the uncertainty. The places where the evidence thins out and honest scientists shrug and say, "We don't know yet," are often the most revealing places of all. They remind us how much of the human past is still buried, still silence, still waiting. So settle in. The first piece of this story begins with a cave in the Altai Mountains of Siberia. The year is 2008. A team of Russian archaeologists is excavating a site they've worked for years, and deep in one layer of sediment, they find something small enough to overlook entirely. The Altai Mountains sit at the meeting point of Russia, China, Mongolia, and Kazakhstan. It's one of those places where geography creates a kind of crossroads, passes through the mountains that animals and humans both used as corridors for migration, river valleys that offered shelter, highlands that could trap a population long enough for evolution to do interesting things. Denisova Cave is cut into the side of a limestone cliff above a small river in that region, and it's been used on and off for an almost incomprehensible stretch of time. The layered sediments inside record roughly 300,000 years of occupation. Not continuous human habitation, not always humans at all, but accumulations of charcoal and bone and stone and tooth and ash that build up the way tree rings do, each layer a frozen moment waiting. Russian archaeologists had been working the site for decades before the find that changed everything. It wasn't glamorous work. Excavating cave sediments is meticulous and slow. You sieve and sort and catalogue. Most of what you find is animal bone, flint flakes, charcoal. The cave had already yielded interesting artifacts, some sophisticated stone tools, an unusual bracelet made from chlorite that still puzzles researchers. But the sediment kept its biggest secret until 2008, when a Russian team working in layer 11 of the East Gallery recovered a piece of bone so small it almost wasn't noticed. It was a distal phalanx, the end segment of a finger. Based on its size, it probably came from a young girl, likely between 5 and 13 years old. The bone was fragmentary, not much larger than a fingertip itself. There was nothing about its physical appearance that shouted discovery. No unusual shape, no striking features. By anatomy alone, you couldn't have told it apart from a modern human child's finger bone without a great deal of uncertainty. And that, it turns out, was the first clue that something unusual was happening, though no one knew it yet. A molar came from the same site, then another tooth years later. These teeth were strange in ways you could actually see. They were large, much larger than both modern human and Neanderthal teeth, with a root configuration that looked more ancient, more robust. If you had only the teeth and none of the DNA, you might have classified them as belonging to something far older than they were. Maybe Homo erectus or another archaic lineage. The mismatch between the seemingly unremarkable finger bone and the anatomically unusual teeth made the picture harder to read, not easier. The physical remains were sending mixed signals. The key came from inside the bone itself. Researchers extracted ancient DNA from the finger fragment and sent it to Svante Pääbo's lab at the Max Planck Institute for Evolutionary Anthropology in Leipzig. This was the same team that had been revolutionizing ancient DNA research, the same group that would later sequence the Neanderthal genome and fundamentally change how we understood human prehistory. They were used to working with degraded, contaminated, difficult material. But what they found when they sequenced the mitochondrial DNA from that Denisovan finger fragment was something they hadn't anticipated. The sequence didn't match modern humans. That much wasn't entirely surprising. But it also didn't match Neanderthals, who had been living in roughly the same part of the world during overlapping time periods. It was distinct from both, diverging from the modern human and Neanderthal lineages at a point roughly twice as far back as Neanderthals diverge from us. The mitochondrial signal suggested this individual's maternal lineage had split from the one leading to modern humans and Neanderthals somewhere around 800,000 to 1 million years ago. This was not a variation on something already known. This was something else. When the nuclear genome was eventually sequenced, the picture shifted somewhat. Nuclear DNA, which captures the full scope of both parents' lineages, showed that Denisovans and Neanderthals were actually sister groups, more closely related to each other than either was to modern humans. The mitochondrial divergence was deeper, possibly the result of an earlier episode of interbreeding with another archaic lineage that contributed mitochondrial DNA to the Denisovan population. So even the initial signals were complicated, carrying echoes of other meetings, other histories layered inside them. What the researchers had on their hands was a population that had never been identified before. Not from bones, not from fossils, not from any artifact previously assigned to a known group. The name Denisovan came simply from the cave, Denisova. In Russian, the cave takes its name from a hermit named Denny, who is said to have lived there in the 18th century. An 18th century Russian hermit now lends his name to an ancient human lineage. History has a sense of humour, but here's what's important to understand about what Denisovan meant in those early years. It was primarily a genetic designation, not a biological or anatomical one. The word described a distinct lineage, a branching point in the human family tree, a coherent genetic signal. It was not a species description in the traditional sense, because there wasn't enough physical material to define a species. There was no skull to study, no postcranial skeleton, no face. There was a finger bone, a couple of teeth, and a genome. Calling them Denisovans was essentially saying, we can see in the DNA that these individuals belong to a group separate from any we previously knew about, and they lived in this cave in the Altai Mountains, so we'll name them for the place. That might sound like a technicality, but it matters enormously for how we interpret everything that comes afterward, because the genetic signal for Denisovans eventually turned up not just in Siberia, but in modern populations thousands of miles away, in people from Papua New Guinea and the Philippines, in Tibetans, in Aboriginal Australians, in indigenous people across island Southeast Asia. The label that began as a place name for a cave became a category that had to expand far beyond that cave, tracking a group or possibly multiple related groups across an enormous span of time and territory. The initial scientific reaction was a mixture of excitement and genuine unease. On one hand, it was a spectacular find, unambiguous evidence of a previously unknown human lineage. On the other hand, it was deeply uncomfortable for anyone attached to cleaner models of human prehistory. The standard picture, Africa, then westward expansion of Neanderthals, then the eventual rise and spread of modern humans, already had enough loose ends. A third major group threw new tangles into every part of it. When did Denisovans split from the Neanderthal lineage? Where did that split happen? How widely did they range? How many times did they meet and interbreed with other groups? Were they one population or several? None of these questions had easy answers, and most of them still don't. Some researchers pushed back on the completeness of the picture almost immediately. Ancient DNA, particularly mitochondrial DNA, can be misleading. It captures only one line of descent. Contamination is a constant risk in ancient specimens, especially bones from cave sites that have been handled by generations of researchers. The Max Planck team had extraordinary protocols to control for these risks, but the scientific community was right to scrutinize the findings carefully. That scrutiny over the following years only deepened the strangeness of what had been found rather than resolving it. Additional sequencing, comparisons with other ancient genomes, analysis of modern populations all kept pointing to the same conclusion. There really was a distinct lineage here. The signal was real. What made the whole thing philosophically striking, not just scientifically interesting, was what it implied about everything we hadn't found. If a fragment of finger bone from a cave in Siberia could reveal an entirely unknown branch of the human family, what else was out there in sediment we hadn't yet sieved? How many other groups had lived and died without leaving any physical trace substantial enough for us to classify? The Denisovans hadn't been hiding. We just hadn't had the tools to see them until the DNA revolution gave us a kind of sight we'd never had before. That's the deepest thing the cave discovery did. It didn't just add a name to the family tree. It made the whole tree look provisional, like a rough sketch on paper that someone had been treating as a finished blueprint. Suddenly the gaps in the fossil record weren't just gaps, they were potential homes for entire lineages. The human past became larger and stranger and more crowded than it had been the day before. So here is where things stood after that initial discovery. Scientists had a genetic signal from one small cave in the Altai Mountains, representing at least 2 individuals, possibly more based on later finds. They had teeth that looked archaic, a finger bone that looked almost modern, and a genome that told a complex story about deep divergence and ancient interbreeding. They had a name for a group whose range they couldn't yet determine, whose appearance they couldn't describe, and whose culture they could only glimpse through artifacts whose ownership was uncertain. Lots of DNA, very few bones, an enormous territory left to map, and questions that would take another decade and a half of work to even partially answer. The cave had given up its secret, but the secret turned out to be not an answer, but a door, opening onto a much larger darkness that scientists are still walking into now. If we're going to let a few fragments of bone and tooth redraw the human story, we have to talk about how that story is being written. Because none of this, Denisovans, ghost ancestors, deep splits, comes straight out of the ground. It comes out of molecules, statistics, and a frankly absurd amount of cleaning. Ancient DNA work starts long before any sequencing machine turns on. It begins with trying to coax molecules out of things that have no biological softness left. A finger bone that's been frozen in cave sediment for tens of thousands of years is more rock than flesh. The collagen is mostly gone. The fats are gone. What remains of the DNA is broken into tiny fragments, riddled with chemical damage. To get at it, scientists drill or scrape a small amount of bone or tooth powder from the interior of the specimen, away from surfaces touched by human hands, animal mouths, or environmental microbes. That powder is then soaked in solutions that dissolve bone minerals and proteins, but leave behind the slivers of DNA. Those fragments are captured, cleaned, and chemically treated to repair some of the damage and make them attachable to the adapters that modern sequencing machines require. By the time the DNA makes it into a sequencer, it's a chaotic mix. Tiny bits from the ancient individual you care about, and vastly more bits from bacteria, fungi, and whatever else has been living in that sediment for thousands of years. Sometimes, if things went badly at any earlier stage, there's also DNA from archaeologists, curators, or the lab staff themselves. The job is not to sequence the Denisovan genome sitting there intact in the bone. The job is to sift a haystack of molecules, most of which are not Denisovan, until you can extract a signal that is. That is why contamination is the ghost that haunts every ancient DNA lab. Modern human DNA is particularly dangerous. It's plentiful, it's everywhere, and if it gets into an ancient sample, it looks very similar to what you're trying to study. You can't just look at a sequence and say, "Ah yes, this is obviously from Dr. Müller who handled the bone last year. It all looks human." So the labs fight back. Ancient DNA work is usually done in rooms that are physically separated from any place where modern DNA is handled. Positive air pressure pushes air out rather than sucking it in. Surfaces, tools, and even the samples themselves are regularly exposed to bleach or UV light to destroy stray DNA clinging to the outside. Researchers wear full-body suits, masks, face shields, double gloves. The scenes look more like a high containment disease unit than a history project. And then, after all of that, the real authentication begins in the data itself. Ancient DNA has a distinctive damage pattern. Over time, certain bases, especially cytosines, tend to break down in characteristic ways, often turning into thymanes near the ends of fragments. When scientists sequence a putative ancient genome, they look for this damage signature. A sample rich in long, undamaged fragments that look like fresh DNA is not a triumph. It's a warning sign. It might mean what they've sequenced is mostly modern contamination. They also look at fragment length. Ancient DNA is short, usually under 100 base pairs, often much less. Modern contamination tends to show up as longer fragments. When a dataset shows the expected mix of short, heavily damaged fragments consistent with its age, confidence grows that the genome really is old. There are other checks. Labs often sequence the people working with the samples, so they can identify and remove any reads suspiciously similar to staff genomes. They repeat extractions from different parts of the bone. They compare results from different labs when possible. In the Denisovan case, the fact that this new lineage was so clearly distinct from both modern humans and Neanderthals while still fitting into the broader hominin tree was itself part of the authentication. Contamination usually pulls an ancient sample toward modern genomes, not away from them into a new branch. Even once you trust that the DNA is genuinely ancient, you're not done. You have to decide which parts of it you're going to pay attention to. There are two main categories here: mitochondrial DNA and nuclear DNA. Mitochondrial DNA, mtDNA, lives not in the cell nucleus but in the mitochondria, the tiny energy factories inside our cells. Each cell has many mitochondria, and each mitochondrion carries multiple copies of its own little circular genome. That means mitochondrial DNA is present in much higher copy numbers than nuclear DNA. When you're dealing with a severely degraded bone, that abundance is a lifesaver. You're more likely to recover an mtDNA sequence than a reasonably complete nuclear genome. But mtDNA comes with limits. It's inherited almost exclusively down the maternal line. Your mitochondrial DNA is effectively your mother's, and her mother's, and her mother's, back in time. It doesn't recombine with paternal DNA. That makes it a clean, simple marker for tracing one narrow line of descent. But it also means it represents only a tiny fraction of your ancestry. It can tell you when two maternal lineages last shared a common ancestor, but it can easily miss other gene flow happening through fathers and through other branches of the family tree. Nuclear DNA, by contrast, is the main archive. It's the 3 billion base pairs in your chromosomes. It comes half from each parent, mixed and shuffled every generation. When scientists talk about sequencing the Denisovan genome, they usually mean nuclear DNA. It's much harder to recover in good quality from ancient material. It's often incomplete, full of gaps and low coverage sections. But even partial nuclear genomes contain vastly more information about relationships, population size, and episodes of interbreeding than mitochondrial DNA ever can. This is why the first Denisovan result, based on mtDNA, suggested an extremely deep split, far back beyond the divergence between us and Neanderthals, while the later nuclear data painted a more nuanced picture. The mitochondria were telling the story of one maternal lineage. The nuclear genome was telling the story of the whole population. Once you have that nuclear DNA, the next step is to ask, where does this genome sit compared to others? Scientists align Denisovan sequences with those from Neanderthals, various modern human groups, and sometimes other ancient hominins. They count differences, how many positions in the genome where Denisovans have one base and Neanderthals or modern humans have another. Because mutations accumulate at roughly predictable rates over long timescales, these differences can act like a clock. But it's not a simple stopwatch. Different parts of the genome evolve at different speeds. mutation rates can vary between populations and over time. So instead of one neat number, you get ranges, models, best estimates plus error bars. When you hear that Denisovans and Neanderthals split around 400,000 to 450,000 years ago, or that the mitochondrial lineage may have diverged closer to 800,000 years ago, those are not exact dates. They're results of fitting the observed genetic distances into models of population history that include mutation rates, past population sizes, and known archaeological dates as anchors. Change the assumed mutation rate, and your divergence times shift. Add a new ancient genome, a Neanderthal from a different region, say, or another Denisovan from a much earlier or later layer, and you may find that your old model no longer fits all the data well. The clock gets adjusted. Sometimes it gets recalibrated entirely. This is why dates in human evolution papers move around over the years. It's not that scientists keep getting it wrong and then finally right. It's that each new piece of evidence allows them to refine their models. The uncertainty ranges are not a flaw. They're an honest admission that we are looking at shadows and trying to infer the movement of bodies. And sometimes the shadows belong to people whose bones we have never seen. When geneticists compare genomes from modern populations and known ancient hominins, they occasionally find segments that don't match any of the reference groups, sections of DNA in modern humans that are too divergent to have come from Neanderthals, too divergent to be simply modern, but also absent from the Denisovan sequences we've sampled so far. Statistical methods can detect these anomalies as signs of ghost populations, ancient human groups that must have existed and interbred with our ancestors, but that have not yet been identified in the fossil record or through direct DNA sampling. They leave no skulls, no named species, just echoes in the genomes of their descendants. Some of the odd patterns in Denisovan DNA, the strange mitochondrial lineage, hints of even deeper admixture, suggest that Denisovans themselves may carry genes from one or more of these ghost groups. In turn, modern humans in some regions carry echoes of Denisovans, and perhaps echoes of whatever had already mixed into Denisovans before that. So when we talk about Denisovans as if they were a single, neatly bounded population, we're smoothing over a reality that the DNA keeps hinting is much messier. Branches within branches. Lineages that meet, mix and vanish. Dates that slide as new methods and new genomes come online. The cave gives us bones. The lab gives us sequences. The sequences give us models, and the models, if we're careful, give us stories. But every step in that chain comes with noise, with risk, with the possibility that what we think we see is only part of the picture. Keeping that in mind matters, because once you accept that Denisovans were first recognized as a pattern in damaged DNA, not as a row of skulls on a museum shelf, it becomes easier to imagine what else might be out there. Other patterns, still buried in sediments and in our own cells, waiting for us to learn how to hear them. So when you hear Denisovans, it's tempting to picture a single group, in a single cave, living a single kind of life. One tiny finger bone in Denisova Cave, one molar, one genome, one population. A tidy label pinned to a tidy box. But evolution doesn't work in tidy boxes, and a single cave has never been enough to stand in for an entire kind of human. Think about our own species. If all you knew about Homo sapiens came from one graveyard in one modern city, you would miss almost everything. You wouldn't see hunter-gatherers in the Arctic. You wouldn't see horticulturalists in the Amazon. You wouldn't see desert herders, rice farmers, office workers, or monks. You would have one thin slice, and you'd be trying to build a whole story out of it. That's where we are with Denisovans. Denisova Cave is important, but it's also an accident of good preservation, cold temperatures, the right sediments, and the right people deciding to drill into a small bone and check the DNA. If the wind had blown a little differently 40,000 years ago, the finger that became the Denisovan might have rotted in the open instead of being sealed in cave sediment. We might still have Denisovans living in our genomes, but we'd be calling them something else, or not calling them anything at all. Everything we've learned since that first genome has been quietly pushing us away from the idea that Denisovan names a single homogeneous population. The genetics are the first clue. When researchers compare Denisovan DNA to modern humans in Asia and Oceania, they don't see one simple pulse of admixture, one moment where Denisovans mixed with us and then disappeared. They see layers. Different modern populations carry Denisovan-like segments that don't all match the Denisova Cave genome equally well. Some people, especially in parts of Melanesia and New Guinea, carry Denisovan-related DNA that's quite close to the Siberian Denisovan sequence we've sampled. Others, including some East Asian populations, carry segments that look more distantly related, still Denisovan-like, but branching off earlier, as if they came from cousins rather than siblings of the cave population. Statistical models that try to fit all this variation do not do well with one neat Denisovan population, branching away from Neanderthals and then sitting there until we meet them. To make the numbers work, you usually need multiple Denisovan lineages, deep substructure within what we call Denisovans, different branches separated for hundreds of thousands of years, occasionally meeting each other and occasionally meeting us. It's a bit like discovering that the Neanderthals who left DNA in Europeans are not genetically identical to Neanderthals from, say, Croatia or Spain, which is exactly what we see on the Neanderthal side. Regional variation, local histories, internal diversity, all hiding behind one convenient name. Neanderthals are a good comparison here because we actually have dozens of their bones from across their range. When scientists sequence Neanderthal genomes from different regions and ages, they find that Neanderthals in Siberia are not quite the same as Neanderthals in Western Europe. There were distinct subgroups, some more isolated, some more connected. Populations that shrank, expanded, merged, and vanished. No one seriously thinks a Neanderthal in Spain was genetically identical to a Neanderthal in the Caucasus, or that their cultures were frozen copies of each other. We understand that Neanderthal is a label for a broad, internally varied cluster of related populations. With Denisovans, we don't yet have the bones to see that variation directly, but the DNA patterns are pointing to a similar kind of spread. When researchers look at Denisovan segments inside modern genomes, they sometimes find that different chunks of Denisovan-like DNA within the same person trace back to different Denisovan sources. One piece looks closer to the Denisova Cave genome. Another piece looks like it comes from a lineage that split from that cave genome hundreds of thousands of years earlier. This is where the term deep substructure comes in. It means that long before we ever met them, Denisovan-related populations had already been diverging from one another for a very long time, probably scattered across a vast area of Asia and maybe beyond. They weren't a single band huddled in a Siberian shelter. They were a network, branches of a tree that had been growing apart, while still occasionally sharing pollen. Some models suggest at least two major Denisovan branches, possibly three. One that contributed heavily to island Southeast Asia and Oceania. Another that contributed mostly to mainland Asia. Maybe a third, more ghostly group hinted at in certain genomes, that doesn't quite match either the known Denisovan sequences or Neanderthals. When geneticists run their simulations, they can sometimes tell that a particular Denisovan-like segment in modern humans came from a population that had already been separate from the Denisova Cave lineage for a long time, longer than many regional differences we see among Neanderthals. That's a strong hint that the label Denisovan is doing a lot of work, covering several related but distinct populations. It's also possible that some of what we are calling Denisovan today will, in the future, be split into more precise categories. Not different species necessarily, but different regional clusters, different traditions, different histories. Think of how we talk about modern humans, East Asian populations, indigenous Australians, Inuit, San hunter-gatherers. All clearly Homo sapiens, all clearly related, but each with their own deep history and local adaptations. If a far future archaeologist had only one Homo sapiens genome from one cemetery, and then tried to explain all living humans with that one reference, their models would start to break the same way our Denisovan models are starting to strain. The hints of multiple Denisovan branches also help make sense of something else: the strange signals of even older admixture inside Denisovan genomes themselves. Some analyses suggest that Denisovans, before we ever met them, had already mixed with yet another, even more ancient human group, one of those ghost populations with no fossil face. If that's right, then different Denisovan lineages may carry different amounts of this older ancestry, depending on where and when they met that ghost group. So when we say that certain modern peoples carry 5% Denisovan DNA, that number is already a simplification. Inside that 5% are threads from multiple Denisovan-related populations, plus echoes of whoever those Denisovans had already met before us. Layers of encounters embedded inside one label. Viewed from this angle, Denisovan stops looking like a species in the strict museum drawer sense and starts looking more like a convenient nickname for a sprawling, time-deep family of related populations that shared a common origin with Neanderthals, then spun out across an enormous territory. Some of those populations were probably living in cold, high-latitude environments like the Altai, adapted to long winters, scarce game, and the peculiar challenges of Pleistocene Siberia. Others may have been thriving in tropical forests, on island chains, along coasts and rivers far to the south. Their tools, diets, social structures and bodies could have been quite different, even while their genomes would still show them as part of the broader Denisovan cluster. We already suspect this kind of ecological spread for Neanderthals and early Homo sapiens. There is no reason to think Denisovans were exempt. The climate shifted, sea levels rose and fell, glaciers advanced and retreated, populations moved, split and reconnected. Some lines died out. Others gave rise to people we would recognize as cousins. From the point of view of genetics, what ties Denisovans together is not that they were one thing in one place at one time. It's that they share a particular position in the human family tree, a sister lineage to Neanderthals, carrying certain distinctive patterns across their genomes and leaving behind distinctive traces in ours. From the point of view of history, the more interesting question is not what were the Denisovans, as if there were a single answer, but how many different ways could Denisovan-related peoples live, adapt, and become part of other human stories? How many climates did they learn to survive in? How many neighbours did they meet? The safest takeaway for now is this. When we talk about Denisovans, we are almost certainly talking about more than one population. Maybe more than one culture, possibly more than one long-lived tradition of tools, myths and adaptations layered across time and space. The first picture we got, the lonely Siberian cave, the anonymous finger bone, the single unexpected genome, was never wrong. It was just too small. As more data come in, the outline is expanding, spreading east and south, branching backward in time. Denisovan identity is turning out to be broader, more complex, and more intertwined with other humans than that early cave-bound image ever suggested. And once you accept that Denisovans were not a single frozen snapshot, but a shifting continent-wide mosaic, another question opens up quietly in the dark. Where, exactly, did they all live? And how far did those branches really reach? To answer that, we have to leave the safety of the cave and follow the DNA. If you look only at fossils, Denisovans are basically a Siberian rumour. A finger bone, a few teeth from the Altai, maybe a jaw from the Tibetan plateau that probably belongs to their side of the family. A handful of fragments scattered across a vast map. But if you look at living people, the centre of gravity shifts dramatically south and east. When geneticists scan modern genomes for Denisovan-like segments, they don't find the highest levels in Siberia or even in northern Asia. Those northern populations often carry a little Neanderthal ancestry, and in some cases a tiny Denisovan signal, but it's faint, like the echo of a distant conversation. The loudest Denisovan echoes show up thousands of miles away in places like New Guinea, parts of Melanesia, and some island Southeast Asian groups. In some of these communities, 5% or more of the genome can be traced back to Denisovan-related sources. Not one source, as we've seen, but multiple, layered Denisovan lineages. It's as if the genetic spotlight that first flickered on in a Siberian cave suddenly swings south, illuminating a chain of islands and tropical coasts. This creates a puzzle. Why is the clearest Denisovan signal in Oceania, when the only unambiguous Denisovan bones we've found are in the cold, dry mountains and caves of northern Asia? There are a few possible answers, and each one hints at a different Denisovan history. One simple possibility is a northern origin with a southern expansion. In this model, Denisovan-related populations emerge somewhere in northern Eurasia after splitting from the Neanderthal line. Over time, some branches head east and south, following coastlines, rivers, or herds into what is now China, Southeast Asia, and ultimately the islands leading toward New Guinea and Australia. Along the way, they meet different waves of Homo sapiens leaving Africa. Some encounters are brief, leaving only small traces in mainland Asian populations. Others are longer or more intimate, producing the large Denisovan ancestry fractions we see in people of Papuan and some Philippine and Indonesian backgrounds today. In this view, the Altai Denisovans are not an isolated oddity. They're just one northern branch of a much larger, mobile Denisovan world. The southern signals in living humans would then be the fossil record we haven't dug up yet. Another possibility is more radical. Maybe Denisovan-related populations had a long-standing presence in the south from very early on. In that scenario, as soon as the Denisovan Neanderthal lineages parted ways, some of those Denisovan ancestors pushed into warmer latitudes and stayed there, evolving in parallel with their colder climate cousins. North and south would both be Denisovan territory, but with deep internal differences. The Altai Cave would merely sample one cold-adapted branch, while the DNA in modern Oceanians would preserve the ghosts of tropical Denisovans we haven't yet met in the ground. Distinguishing between these models is hard, because the evidence is patchy and heavily biased. Cold, dry places are kind to bones and DNA. Warm, wet, tropical places are not. In a Siberian cave, a finger bone can sit in cool sediment for tens of thousands of years, shielded from heat, water, and most microbes. The DNA breaks down slowly, proteins linger. If we're lucky, we can still read the genome like a book, page after page. In a lowland jungle or a humid island forest, a bone dropped on the ground is an invitation to decomposition. Heat speeds up chemical reactions that shred DNA. Water percolates through sediments, moving and dissolving fragile tissues. Microbes and insects are everywhere, eager to recycle every trace of collagen and fat. Even if a bone survives physically, its genetic material can be scrambled beyond recognition in just a few thousand years. That means our fossil and DNA map of ancient humans is not a neutral photograph of where they lived. It's a distorted image, sharpened in the cold and blurred in the heat. When we say, we only have Denisovan bones from the Altai and Tibet, that might not mean Denisovans preferred mountains and caves. It might just mean mountains and caves have been kind enough to keep a few of their bones intact. Archaeology has similar blind spots. Stone tools survive well, but stone is not labelled with species names. If you find a flake or a hand axe in a Southeast Asian cave from 200,000 years ago, and you know that both archaic humans and early Homo sapiens were in the region at various times, it's very hard to say who made what without DNA from the bones nearby. There are, in fact, several fossil and archaeological candidates across Asia that people have quietly wondered about in light of the Denisovan discovery. A robust jaw from Jahe on the Tibetan plateau, lacking a clear Homo sapiens signature, turned out to carry proteins that fit better with the Denisovan Neanderthal side of the family than with us. It's not a full genome, but it's a strong hint that Denisovan-related humans were living at dizzying altitudes long before modern Tibetans. In China and Southeast Asia, there are late archaic human fossils that don't slot neatly into Neanderthal or Homo sapiens categories. Skulls and jaws from places like Shujiayao, Zhuchang, or some of the remains in Laos and other sites show mosaics of traits, some archaic, some more modern looking. Different researchers have put different labels on them over the years, from archaic Homo to various regional names. None of them are securely Denisovan. But once you know Denisovans existed, it's hard not to wonder whether some of these faces belong to their broader clan. There are also tool traditions in Southeast Asia and island regions that persist for long stretches while similar technologies elsewhere are changing more rapidly. Some scholars have floated the idea that at least part of this enduring pattern could reflect long-lived local populations of archaic humans, possibly including Denisovan-related groups, maintaining their own ways of doing things alongside incoming Homo sapiens. For now, these links are suggestive rather than conclusive. Without ancient DNA or very clear anatomical markers, we're left with maybes and could-bes. But the pattern that emerges is consistent with the genetic story. Denisovan-related humans were not confined to one Siberian locality. They were spread in some form across a wide Asian canvas. That brings us back to Oceania, where the Denisovan signal is loudest. If populations ancestral to modern Papuans, indigenous Australians, and some island Southeast Asian groups picked up a large dose of Denisovan-related DNA somewhere along their journey, that implies Denisovan-related peoples were present in or near those regions at just the right time. One possibility is that the encounter happened on the way, somewhere on mainland Southeast Asia or the Sunda Shelf, before people crossed the now submerged land bridges and straits towards Sahel, the combined Ice Age continent of Australia and New Guinea. Another is that parts of what we now think of as Denisovan ancestry actually represent multiple archaic groups in the broader region that our tools can't yet fully disentangle. Different branches of Denisovan-related peoples may have occupied different islands and coasts, each contributing a slightly different genetic flavour to the modern populations that moved through or settled there. Either way, the fact that Denisovan-like ancestry peaks in populations that historically lived in tropical forests and island archipelagos forces us to expand our mental image. Denisovans were not just cave dwellers in the north. At least some of their lineages were probably comfortable with monsoon cycles, dense vegetation, coastal resources, and the challenges of island life. This has implications for how we think about their abilities. To reach certain islands, even during periods of low sea level, ancient humans would have needed some form of watercraft and navigation, however simple. The genetic evidence strongly suggests that Denisovan-related populations interacted with the ancestors of people who went on to settle remote islands. That doesn't prove Denisovans themselves were sailors, but it keeps the door open to the idea that they, like us, and maybe like some Neanderthal groups, were capable of crossing water barriers when conditions and motivations aligned. So where did Denisovans live? Based on what we know now, the safest answer is across much of Asia, from high, cold plateaus and northern caves to warmer lowlands and, indirectly at least, the gateway islands leading toward New Guinea and Australia. The densest direct evidence still clusters in the north, in places where permafrost and limestone caves have preserved bones and molecules for us to find. The strongest genetic echoes in living people are in the south and east, in regions where the ground is far less generous to ancient remains. Between those two poles stretches a broad, dimly lit zone of possibility, Central and Eastern Asia. mainland and island, Southeast Asia, perhaps even parts of South Asia. All regions where archaic humans clearly lived, where early Homo sapiens moved and mixed, and where our current fossil record is almost certainly an incomplete sampling of who was actually there. As more caves are excavated, more sediments screened, and more ancient DNA pulled out of seemingly unpromising fragments, the Denisovan map will almost certainly change. New bones might anchor them firmly in Southeast Asia. Proteins from a tooth in India or Sumatra might reveal a southern branch we've only glimpsed in modern genomes. Or we might discover that what we've been calling Denisovan actually hides several long separated lineages spread across different ecological zones. For now, every map you see of Denisovan range should be read as provisional, drawn in pencil rather than ink. A few solid dots in the north, a wide cloud of genetic influence in the south and east, a lot of blank space in between that is probably not truly empty, just archaeologically quiet. What's clear is that when our ancestors walked out of Africa and into Eurasia, they did not enter an empty world. They stepped into a continent already holding multiple kinds of humans, Neanderthals in the west and north, Denisovan-related peoples spread through the east and south, each with their own territories, traditions, and deep local histories. And in many places those histories overlapped. The ranges of Neanderthals, Denisovans, and early Homo sapiens were not neatly separated on some ancient zoning map. They touched, blurred, and crossed. At those boundaries, where territories and lifeways met, something very human kept happening, again and again. They had a few options when they met. They could ignore each other, keeping to opposite sides of a valley, listening to distant voices and watching smoke on the horizon. They could fight, driving one group away from a crucial water source or a good cave. They could trade, cautiously at first, then more confidently as trust or sheer habit built up over generations. And sometimes they could do something that leaves a very particular fingerprint in our DNA. They could have children together. When geneticists talk about Denisovan ancestry or introgression, they're talking about that fingerprint. Traces of DNA in modern people that look so different from ordinary Homo sapiens variation that the simplest explanation is, this piece came from another related human group. In practice, detecting that isn't magic, it's statistics. Modern human genomes are mostly very similar to each other. There are differences, of course, but they tend to form recognizable patterns. African variation, European variation, East Asian variation, and so on. When you line up genomes from many people and compare them, most regions of the DNA fall into these broad, familiar patterns. Then there are the weird stretches, segments that don't fit cleanly into any modern cluster. Segments that match almost exactly to the DNA we've extracted from Neanderthal bones or to the Denisovan genome from Denisova Cave. Scientists scan genomes from living people, searching for these outliers. In some populations, they find long blocks of sequence that are nearly identical to Neanderthal DNA. In others, especially in parts of Asia and Oceania, they find blocks that match the Denisovan genome much more closely than they match any modern human reference. Those blocks are like fossils written in code. They're the genetic echo of past matings between different human groups, introgression events, where genes jumped species boundaries and then rode down the generations inside Homo sapiens lineages. The length and pattern of those blocks tell us about timing. Long, unbroken chunks suggest more recent mixing. Recombination hasn't had time to chop them into tiny fragments. Shorter, more scrambled bits point to older events, where hundreds or thousands of generations of reshuffling have sliced and mixed the archaic DNA into smaller pieces. By looking at which populations carry which kinds of blocks and how those blocks are arranged, scientists can start reconstructing a timeline of encounters. For Denisovan-related DNA, that timeline is not a single moment. It's more like a series of overlapping ripples. One major signal shows up in people from New Guinea, parts of Melanesia, and some island Southeast Asian groups. In these populations, there are relatively large amounts of Denisovan-like DNA organized in ways that point to a substantial interbreeding event, or perhaps several closely spaced events, somewhere along the route towards Sahul, the ancient continent combining Australia and New Guinea. Genetic modelling suggests this mixing probably happened after the ancestors of these groups had already split away from the ancestors of most mainland Asians, but before they fully reached and settled the Australian-New Guinean landmass. That would place the encounter somewhere in Southeast Asia or on the now submerged continental shelves that once linked many of today's islands. The Denisovan segments in these populations also look, in some respects, quite different from the Denisovan-like DNA that shows up in East Asians. That's a clue that we're not dealing with a single uniform Denisovan population, but with at least two deeply diverged branches. In East Asia and parts of mainland Southeast Asia, there is a second, distinct Denisovan signal. The archaic DNA chunks there don't line up perfectly with the segments seen in Papuans and Melanesians. They appear to come from a Denisovan-related group that had split from the branch, contributing to Oceanians long before modern humans ever encountered either of them. This means that at least two separate Denisovan populations, far apart in their own evolutionary history, met and mixed with different waves of Homo sapiens at different times and places. Some studies even hint at more complexity, perhaps three or more Denisovan-related lineages, each leaving a slightly different imprint across Asia and Oceania. The details are still being debated, the models refined and revised. But the broad picture is clear. Interbreeding with Denisovan-related peoples was not a one-off event. It was a repeated theme as modern humans moved through Eurasia. Layered on top of this is the Neanderthal story. Our species carries Neanderthal DNA almost everywhere outside Africa, a legacy of early encounters in the Middle East and possibly additional episodes in Europe and Central Asia. Denisovans themselves carry Neanderthal DNA, and at least one Denisovan individual from Denisova Cave, the famous Denny whom we'll meet in the next section, was the direct child of a Neanderthal mother and a Denisovan father. Taken together, these patterns don't look like three neat, separate branches of a family tree, heading in different directions and never touching again. They look like a braided river. Neanderthals, Denisovans, and Homo sapiens were distinct lineages, yes, with their own histories and adaptations. But where their ranges overlapped in time and space, they were capable of producing fertile offspring. Genes flowed across those boundaries, not constantly, not everywhere, but often enough to leave a lasting mark. It's important to be careful about what that does and doesn't mean. Interbreeding does not imply some timeless, peaceful harmony between species. A few % of archaic ancestry in a genome could come from relatively rare events spread over thousands of years. A Neanderthal modern human child here, a Denisovan Homo sapiens child there, in small frontier zones where groups pressed against each other. Those contacts could have been friendly, violent, or anything in between. They could have involved kidnapping, alliance marriages, coerced unions, or genuine partnerships. The DNA can tell us that genes moved. It cannot, by itself, tell us the emotional texture of those moments. What gene flow does imply is proximity. For Denisovan-related DNA to end up in modern Papuans, Denisovan-related people had to be living close enough to the ancestors of Papuans for mating to occur. For East Asians to carry a different Denisovan-like component, there had to be another Denisovan-related group, somewhere in their own migratory corridor at the right time. When models suggest that Neanderthal and Denisovan lineages themselves exchanged genes, that means there were places perhaps in Central Asia, perhaps in the Altai, where Neanderthal territories and Denisovan territories overlapped, not just in space, but in years and centuries. Individuals from those groups met, interacted, and sometimes had children. Each of these events is a point on the map where two ranges blur. Each is a moment where the clean lines on our diagrams dissolve into messy human reality. From the perspective of survival, interbreeding was not just an accident, it was a potential strategy. Imagine you're part of a small Homo sapiens group moving through a landscape you've never seen before. The plants are unfamiliar, the seasons feel wrong, the animals behave in ways you haven't fully learned. You carry knowledge from your ancestors, but that knowledge was honed in African savannas or woodlands, not in Siberian forests or Southeast Asian islands. Now imagine that in this new place, you encounter another kind of human. They've been here much longer, their bodies and genes are tuned over hundreds of thousands of years to this particular climate, this altitude, these parasites, these foods. Most of the time you and they might keep your distance. You might compete for game or trade obsidian for shells, or simply watch one another warily across the river. But if, sometimes, children are born between your group and theirs, those children carry something precious, a combination of your flexibility and their deep local adaptation. Some of the Denisovan-derived genes we see today seem to have done exactly that. Variants affecting oxygen processing help modern Tibetans live and work at high altitude. Certain immune-related genes from archaic humans may help modern populations fight off pathogens they did not encounter in Africa. Other Denisovan segments may have influenced fat storage, skin physiology, or how the body responds to certain environmental stresses. Most archaic DNA that entered the Homo sapiens gene pool probably washed out over time, neutral or slightly harmful in its new context. But the segments that helped, even a little, had a better chance of sticking around, being passed on and spreading. In this sense, interbreeding was another tool in the survival kit. Not one our ancestors consciously planned as a biological strategy, of course, but one that natural selection could work with. When conditions changed, any gene that gave an edge, no matter where it originally came from, could be woven into the tapestry of our species. The emerging picture, then, is not of isolated human species evolving on separate stages, but of a loose, shifting network. Neanderthals anchored the west and north. Denisovan-related groups occupied wide swaths of the east and south, and Homo sapiens expanded outward from Africa, threading through both territories. At the margins where these groups met, genes crossed back and forth, sometimes once, sometimes many times, leaving faint but detectable signatures that we're only now learning to read. For most of history, those signatures were invisible. No one looking at a person in New Guinea or Tibet could have guessed that a sliver of their genome came from a long-vanished people in a Siberian cave. No Neanderthal, staring into the fire beside a Denisovan partner, could have imagined future scientists reconstructing their family story from powdered bone and double helices. Yet that is the story our DNA is telling, of repeated meetings between different kinds of humans, not all friendly, not all frequent, but enough to bind our separate branches together in ways we are still untangling. Sometimes those meetings were recorded in particularly vivid ways, like the life of a single girl who was half Neanderthal and half Denisovan, whose existence would remain unknown for tens of thousands of years, until someone ground a tiny bone from Denisova Cave and read her ancestry in the dust. She's known to us by a nickname, Denny. In life, she wouldn't have had that name. She would have just been a girl in a family, in a band of people moving through the forests and slopes around the Altai Mountains. She might have helped carry wood, gather plants, clean hides. She might have watched older hunters prepare for a long trip, listened to stories told beside the fire. To the people around her, she was not an evolutionary event. She was someone's child. We only know she existed because of a single fragment of bone. It was so small that at first it didn't look promising, just another splinter from the mass of broken animal and human remains that filled Denisova Cave. Too incomplete to tell you age, sex, or species by eye. It might easily have gone into a discard pile. Instead, it went through a screening process. Researchers used collagen fingerprinting, looking at the protein makeup of fragments to pick out those likely to be human. That little piece of bone passed the test. Then it went on to the next stage, ancient DNA extraction. When the genome came back, it didn't just nudge our picture of the past. It snapped it into a new shape. Because Denny, this teenage girl from tens of thousands of years ago, turned out to be something astonishing. A first-generation hybrid. Half Neanderthal, half Denisovan. Not a bit of Neanderthal ancestry way back in the family tree. Not some Denisovan DNA from a distant ancestor. Her genome was split, cleanly and evenly. One set of chromosomes matching Neanderthals, one set matching Denisovans. Her mother, based on the mitochondrial DNA, was Neanderthal. Her father was Denisovan. Genetically, there was no ambiguity. When scientists compared Denny's genome to known Neanderthal sequences, about half her DNA lined up almost perfectly. The other half matched the Denisovan genome from Denisova Cave, itself reconstructed from another individual who had lived there earlier. Even within those halves there were surprises. Her Neanderthal side did not match just any Neanderthal. It looked closer to Neanderthals from western Eurasia than to an earlier Neanderthal who had lived in the same cave long before. That suggests her mother, or her mother's recent ancestors, came from far away, part of a west to east movement of Neanderthal populations. Her Denisovan side looked like the Denisovan genome we already had from the cave, but not identical. Denny's father carried Neanderthal DNA of his own, evidence that there had been earlier mixing between these two groups before Denny was even born. So inside this one individual, we see at least two episodes of contact layered on top of each other. A Denisovan group that had already absorbed some Neanderthal genes. A Neanderthal woman descended from Western populations who found herself in the Altai. And a child whose very existence means that these people were not ghostly presences on opposite hillsides, forever separate. They met closely enough and often enough for at least one Neanderthal woman and one Denisovan man to share a life long enough to conceive and raise a daughter. What does that mean in practical terms? It tells us the Altai region was not just a boundary. It was a contact zone. Denisova Cave itself has yielded both Neanderthal and Denisovan remains from different layers and different times. The cave was not the exclusive home of one species. It seems to have been used intermittently by both, over tens of thousands of years. Neanderthals were there. Denisovans were there. At least once, we know they were there at the same time, or close enough in time for lineages to overlap. Denny crystallizes that overlapping into a single life. She implies something about how these populations used the landscape. If Neanderthals could move into the Altai from the west, and Denisovans could hold territory there from the east or south, this region was a kind of hinge, where ranges met and sometimes interlocked. Think of it as a long, slow dance of expansion and retreat. In colder phases, Neanderthal-adapted groups might have pushed into the area, tracking the animals and plants they knew, exploiting the caves and valleys that offered shelter. In other periods, Denisovan-related groups might have dominated, tuned to the particular challenges of Central Asian environments, occupying the same caves, using the same springs. Distances in Ice Age Eurasia were not measured in miles but in days of walking, in number of safe camps between water sources in seasonal cycles of game movement. Over thousands of years, those movements would bring different human groups into repeated, shifting contact. Denny is a snapshot of one of those moments when the circles overlapped, but she is also a caution. It is tempting to see her and imagine a valley full of hybrids, a thriving mixed community where Neanderthal accents and Denisovan accents merged into a single voice. We do not know that. So far, she is one data point. An extraordinary data point, unique in its clarity, but still just one. We have no census of the Altai. We don't know whether first-generation hybrids were common there, rare, but accepted, or rare and treated as outsiders. We don't know whether Denny lived into adulthood, whether she had children, whether any of her genes continued forward into later generations. What we can say is that the chance of finding a first-generation hybrid in such a tiny sample argues against extreme rarity. Denisova Cave has yielded only a handful of individuals whose genomes can be read. Among them, we find a girl whose parents came from two different human species. That suggests that Neanderthal Denisovan matings, while probably not everyday events, may not have been a once-in-all-time accident either. In population genetics terms, if you randomly pick a few individuals and one turns out to have parents from distinct lineages, it implies a background level of gene flow. Not a flood, but certainly not a sealed wall. The boundaries between Neanderthals and Denisovans, at least in this region, were porous. And this fits with the broader pattern we see in genomes across Eurasia. Neanderthals and Denisovans both show traces of each other's DNA. Modern humans show traces of both, and of at least two or more Denisovan-related groups. No line is completely clean. No branch is entirely isolated. Instead of thinking of species borders as sharp cliffs, it may be better to picture them as broad, uneven shores. Most of the time, most individuals stay on their own side. But here and there, in particular bays and inlets, the water touches and mixes. The Altai looks like one of those inlets. Denny's genome also nudges us to rethink how stable these populations were. If Neanderthals with Western ancestry could appear in Central Asia, their world was more connected than we sometimes imagine. Their territories were not fixed blocks on a map, but expanding and contracting ranges with waves of migration, replacement and intermingling. The Denisovans using Denisova Cave were not an isolated pocket either. The Neanderthal signal in Denny's father suggests his ancestors, or he himself, had encountered Neanderthals before. That implies either earlier contact zones elsewhere, or a longer history of overlap in the region than a single brief encounter. In other words, Denny is not just one story of one family. She is evidence of a relationship between lineages that had been unfolding long before she was born. And yet, for all that, she also reminds us how much we don't know. We don't know what language was spoken in her camp, or whether Neanderthal and Denisovan groups shared words when they met. We don't know if they had formal rules for who could partner with whom across group lines, or if such pairings were exceptional and fraught. We don't know if her mother missed her western homeland, or if her father had kin in neighbouring valleys. We don't know if other children in her group looked like her, with the same mixed ancestry, or if she stood out as something unusual. The DNA cannot give us those details. It can only whisper here, at this point on the map of time. Two long-separated lineages came together in one body, and that whisper changes the way we read the rest of the record. Before Denny, it was possible to talk about Neanderthals and Denisovans as almost entirely separate, meeting rarely, exchanging a few genes somewhere in the deep background. After Denny, that picture feels too clean. We have to allow for the possibility of long-term overlap in places like the Altai. Not constant cohabitation, but repeated phases of contact, shared hunting grounds, occasional shared children. We have to imagine frontiers not as lines of exclusion, but as gradients. That, in turn, reinforces the deeper theme running through the Denisovan story. The human family tree was never a set of solitary trunks growing straight toward the sky. It was a thicket, with branches crossing and grafting, with pollen drifting from one stem to another. Denny is one of the clearest graft points we've ever seen. A single life where Neanderthal history and Denisovan history are woven together so tightly you can't pull them apart. And she prepares us for the next question, the one that has haunted the Denisovan story from the moment their DNA was first sequenced. If Neanderthals and Denisovans could meet and have children, and if modern humans could do the same with both, what did these other humans actually look like? When our ancestors stood face to face with them, what did they see? If you went back in time and walked into a Denisovan camp, the first thing you'd notice is not what they looked like. It would be the warmth of the fire, the smell of smoke and meat, the murmur of voices, the same small, ordinary human things that fill every gathering place our species has ever made. But the question still lingers when one of them turned and looked at you. What kind of face would you see? Here we run into a strange contrast. On the genetic side, Denisovans are among the best-known ancient humans. We have high-quality genomes. We can trace their DNA in living people across half the planet. Their genetic voice is loud and clear. On the skeletal side, they are almost mute. For Neanderthals, we have whole skeletons, skulls, rib cages, hands, feet. We can see the curve of the brow ridges, the shape of the nose, the angle of the jaw. For Denisovans we have crumbs, a few teeth, a fragment of finger bone, a small piece of long bone, a partial jaw from the Tibetan plateau. Maybe a few more odds and ends, still debated, no complete skull, no full skeleton, not even a whole face. That scarcity matters. Bones are how we normally reconstruct appearance. Skulls tell us about brain size, facial shape, nasal structure. Pelvis and limb bones tell us about body proportions, height, gait. Teeth give us clues about diet, development, even sometimes sex. With Denisovans, we're trying to paint a portrait with almost no canvas. Denisova Cave itself has given us several key pieces. The famous finger bone that first yielded Denisovan DNA is from the last joint of a pinky. By itself, it doesn't tell you much visually. It belonged to a girl, maybe 13 or so, and it looks broadly similar to the same bone in Neanderthals and early modern humans. Nothing obviously alien. Other fragments from the cave include bits of long bone. They hint at individuals that were not tiny, not unusually gracile. But again, there's not enough to say with confidence. They were this tall, or they were built this way. The most informative Denisovan-related fossil so far isn't from Denisova Cave at all. High on the Tibetan plateau, in a cave called Baishia, monks once venerated a large fossil jaw as a relic. Decades later, it was examined by scientists. There was no recoverable ancient DNA, but proteins preserved in the dentine of the teeth carried a Denisovan signal. The jaw is massive compared to most modern humans. Broad, thick-boned with big molars, it looks, at first glance, more like some archaic Asian fossils than like us. No chin. Strong, heavy structure. The kind of jaw you might expect in a population doing a lot of chewing on tough, fibrous foods. But even here we have to be careful. This is one individual from one place at one time. We don't know if he or she was typical. We don't know if high-altitude Denisovans tended to be more robust than Denisovans living in forests or coastal environments. We don't know how much variation there was between different Denisovan-related populations. Still, when you line up the pieces we do have, some cautious patterns begin to emerge. The teeth from Denisova Cave are large, significantly larger than most modern human teeth, and outside the usual range of Neanderthals. They have some distinctive shapes in the roots and crowns that don't match neatly with either group. The Tibetan jaw is big-boned, with those same broad molars. Taken together, they suggest that at least some Denisovan groups were physically robust. Big jaws, big chewing surfaces, probably capable of processing tough diets in demanding environments. But from that to a complete face is a very long leap. This is where new methods come in, trying to fill the space between genes and bones. One approach looks not at DNA sequence itself, but at methylation patterns, tiny chemical tags that help regulate how genes are turned on and off. In living people, certain methylation patterns are associated with particular anatomical outcomes. For example, changes in regulation of genes involved in bone growth can affect the shape of the jaw, the angle of the forehead, the size of the rib cage. Researchers have taken Denisovan DNA and reconstructed likely ancient methylation maps, then compared them to those of Neanderthals and modern humans. From these they infer which genes were more or less active in Denisovans, and what that might have done to their skeletons. It's a little like looking at the sheet music instead of hearing the performance, and trying to imagine how the song would sound. You can get some things right, but you have to translate very carefully. These studies suggest that Denisovans may have combined features we usually think of as Neanderthal-like with others that are closer to, or even beyond, the range of modern humans. Some reconstructions based on this work propose that Denisovans had wide skulls, with large dental arches, a long face, and a fairly broad nose. Perhaps not as projecting a midface as classic Neanderthals, but still strong, with big chewing muscles. They may have lacked a prominent chin like Neanderthals and had robust rib cages and limbs adapted to heavy physical labour and cold climates in at least some regions. Protein analysis adds another layer. In cases where DNA is too degraded, proteins preserved in bone and tooth can still carry evolutionary signals. The Tibetan jaw was identified as Denisovan partly through the comparison of its proteins with those predicted from Denisovan genomes. The presence of particular collagen variants can hint at adaptations to things like altitude, temperature, or diet. This doesn't tell you the shape of an eyebrow, but it reinforces the idea that Denisovans were not delicate, fragile people. They were built to survive in hard places, high plateaus, cold mountains, heavily seasonal environments. On top of this, some researchers use statistical morphology. They take measurements from many known human and Neanderthal skulls, look at how genetic differences map onto anatomical ones, and then ask, if Denisovans occupy this particular place in genetic space, where would their anatomy likely fall? This kind of modelling is powerful, but also abstract. It doesn't give you a photograph. It gives you probabilities that certain features would be more or less pronounced compared to Neanderthals and early modern humans. The most honest answer these methods give is subtle. Denisovans were probably recognisably human in the way Neanderthals were. 2 eyes, a nose, a mouth, a forehead, hair, expressions, gestures. You would not confuse them with another animal. But if you were used to living among early modern humans, you might notice some differences. A heavier jaw, slightly different proportions of face to skull, maybe broader cheekbones in some populations, deeper jaws in others, bigger teeth when they smiled. A build that in some regions tended toward the stocky and powerful end of the human spectrum. And yet, that's only one side of the picture. Because Denisovans were not a single uniform population frozen in a single environment. Their DNA shows up from Siberia to New Guinea, from the Altai Mountains to island Southeast Asia. They lived at 3,000 plus meters on the Tibetan Plateau and at sea level in tropical climates. They interbred with Neanderthals in the north and with the ancestors of modern humans moving through Asia. Over tens of thousands of years, natural selection would have pulled different Denisovan-related groups in different anatomical directions. High-altitude Denisovans might have had barrel chests and particular body proportions to cope with thin air and cold nights. Forest-dwelling Denisovan-related peoples closer to the equator might have been leaner, with darker skin and different hair types. Coastal groups could have developed strong swimming adaptations in lifestyle, if not in anatomy. In other words, there may not have been one Denisovan look. There may have been many. What we tend to see, though, are single images, an artistic reconstruction in a museum, a face staring back from a documentary thumbnail. Strong brow ridge, wide nose, bronze-tinted skin, thick hair. The kind of image that feels solid and definitive. It's worth quietly setting those pictures beside the evidence we actually have. We do not know their skin colour for certain. We do not know their eye colour, hair texture, or facial hair patterns with anything like the confidence we sometimes claim for Neanderthals. Some genes involved in pigmentation and hair show archaic variants, but translating those into exact appearance is still a developing field, full of assumptions. We do not know whether Denisovans had a Neanderthal-like occipital bun at the back of the skull, or a more rounded modern human shape, because we do not have a complete Denisovan skull. We do not know the precise angle of their brow ridges, the depth of their eye sockets, the slope of their foreheads. Every reconstructed face you've seen is an informed guess. A guess grounded in comparative anatomy, methylation-based predictions, and what we know of their environments, but a guess all the same. If anything, the safest mental image might not be a single face, but a crowd. Imagine a campfire circle in Pleistocene Asia. Some individuals tall, some shorter. Some more robust, some a little more gracile. Variations in skin tone, in nose shape, in hair. A range of Denisovan-related faces, overlapping in many ways with early modern humans and Neanderthals, but with their own statistical centre of gravity. If you walked through that crowd, you might sometimes think, he looks like a Neanderthal I saw in a different valley. At other times, she looks almost like the people I came with from the south. And then someone would turn, and something about their eyes, jaw, and cheekbones together would strike you as unfamiliar. Not non-human, just a different way to be human. That, for now, is as close as we can reasonably come. The truth is, our picture of Denisovan appearance is still being drawn, and the pencil is very light. Each new fragment of bone, each new protein sequence, each new statistical model may darken a few lines, sharpen a feature here or there. But until we find a well-preserved skull, or better yet several, the faces we imagine will remain mostly shadows, shaped loosely by teeth and jaw, outlined by genes and climate, filled in by our own expectations. And even when we finally do uncover that skull, it will belong to someone from one place, one group, one moment in Denisovan history. There will still have been others elsewhere, whose faces we may never fully see. Yet, even as their features blur in the distance, one thing remains sharply in focus. They were close enough to us, and to Neanderthals, that when our ancestors met them, they saw not monsters or strangers from another world, but other people. People different enough to notice, similar enough to love, to fear, to live beside, to have children with. Which brings us to another mystery. If Denisovans were so human, so capable, where are their campsites, their tools, their art? If they left their genes in millions of us, why did they leave so little in the ground? To answer that, we have to step away from their bones, what few we have, and look at the stones and ash they might have left behind. If you look at a diagram of Denisovan DNA, it seems bold and clean. Branches, dates, percentages, lines that tell you who mixed with whom and roughly when. If you look at the archaeology, everything smears. The core problem is simple, and it haunts almost every discussion about Denisovans and culture. Caves and campsites are not labelled. Nowhere in Denisova Cave does the sediment politely announce, "This layer was made by Denisovans." Nowhere on a stone tool is there a microscopic engraving that says, "Napped by Neanderthal," or "Made by early Homo sapiens on a Tuesday." The ground records objects, not identities. In Denisova Cave, that becomes a very specific headache, because we know at least 3 different human groups used that same space over tens and hundreds of thousands of years. Neanderthals, Denisovans, and later on, modern humans. Their bones and DNA show up in different layers. Their tools often do not sort themselves so neatly. Archaeology loves clean associations, a skeleton in a layer, a cluster of tools around it, a hearth in clear stratigraphic context, all sealed together by dating methods. When that happens, you can cautiously say, this species probably made these artifacts. But in Denisova Cave, most of the time, we do not find Denisovan bones sitting exactly beside a particular toolset. Instead, we find stone artifacts spread through layers that have, at different points, yielded DNA from different humans. Imagine a long-used mountain lodge. Over centuries, sheep herders, hunters, travellers and families all share the same fireplace. They leave behind knives, cooking pots, bits of clothing. Hundreds of years later, someone digs it up and has to decide which objects belonged to which group, with no photos, no labels, just layers of ash and rubble. That's Denisova Cave on Ice Age mode. So what do we actually see there? The material culture, what people left behind, is mostly stone tools, animal bones with cut marks, and evidence of fire use. In some layers there are ornaments, beads, pendants, objects that look suspiciously like symbolic or decorative items. Some of the stone tools are relatively simple, flakes, scrapers, points. Others show more planning, with prepared cores and standardized shapes. The cave's deep history stretches back almost 300,000 years of intermittent occupation, so the tool styles change over time as environments and populations shift. In a few layers dated to around 50,000, 40,000 years ago, archaeologists have found something especially intriguing: a worked fragment of chloritolite, a greenish stone shaped into what many interpret as a bracelet, with a carefully drilled hole. There are also pierced animal teeth and bone points that look like needles or awls. These finds sit roughly in the same time frame as some of the Denisovan DNA from the cave. It's tempting to leap straight to the headline, Denisovans made jewellery, but this is where that archaeology problem bites back. Those same general time layers also show evidence of Neanderthals, and possibly early modern humans, passing through or using nearby regions. Sediments can be mixed. Fine-scale dating is never perfect. A layer that looks like a single moment may be a palimpsest of occupations stretched over centuries or more. So instead of Denisovans made this, what we can honestly say is softer. Someone in a cave where Denisovans definitely lived, at times overlapping with other humans, made these things. That might sound frustratingly vague, but it's the kind of caution that keeps archaeology from sliding into wishful storytelling. To make sense of this mess, archaeologists lean heavily on three main clues: continuity, sudden shifts, and dates. Continuity means, does a particular style of tools or ornaments show up again and again in layers that, by genetic and dating evidence, seem dominated by the same group? If Denisova Cave had, say, 5 Denisovan-rich layers in a row, all featuring a distinctive toolkit and no sign of other humans, we could be more confident about attributing that toolkit to Denisovans. Sudden shifts mean, does the technology change sharply at the same time that the genetic signal changes? If for thousands of years you have one style of tools, then suddenly a new style appears right when Neanderthal DNA shows up in the sediments, you can suspect that incoming Neanderthals brought their own technological traditions, or at least inspired a cultural revolution. Dated layers are the scaffolding under all of this. Radiocarbon, luminescence, uranium series and other methods tell us when sediments were laid down, within a range of error. This chronology lets us ask whether Denisovan DNA and particular artifacts really belong to the same time slice, or whether we're just seeing a vertical coincidence in the dirt. In Denisova Cave, those 3 lines of evidence only partially line up. There are layers where ancient DNA from sediment points clearly to Denisovans. There are layers where Neanderthal DNA dominates. There are layers with little to no detectable human DNA at all, but plenty of tools. The cultural record seems continuous across some of these zones. There is not always a neat technological border between Denisovan-rich layers and Neanderthal-rich layers. In some periods, both groups may have been using similar regional traditions of stoneworking and ornament production. This is not unique to Denisovans, it's a familiar problem from Neanderthal archaeology as well. For decades, there has been debate about whether Neanderthals invented symbolic behaviours, ornaments, possible art, carefully chosen pigments, or whether they only started using these things after modern humans arrived in Europe and the Near East. In some caves, objects that look like jewellery or personal adornment show up in levels that many researchers still label Neanderthal. But the dating can be fuzzy. Layers can slump, mix, get disturbed by animals, water, or later human activity. A bead that looks like it belongs to Neanderthals might actually have slid down from a later modern human layer. So you get two camps. One says, Neanderthals had their own symbolic culture. The other says, we can't be sure these aren't modern human imports, or at least modern human inspired. The same caution applies to Denisovans, but with even thinner evidence. Those green stone bracelet fragments, the worked bone points, the pierced teeth from Denisova Cave, many scholars see them as signs of complex behaviour, maybe symbolic expression. But were they made by Denisovans, by Neanderthals, by early Homo sapiens visiting or passing through, by some combination of groups sharing ideas? At best, we can assign probabilities, not certainties. Some time intervals in the cave appear, on current evidence, to have only Denisovan DNA. If an artifact is securely dated and embedded in that narrow window, the case grows stronger that Denisovans made it. If the same style of ornament shows up across multiple Denisovan-rich layers, with no overlap from other human DNA, confidence increases again. Yet the record remains patchy. We do not have dozens of Denisovan-only sites scattered across Asia, each with the same distinctive toolkit. We have one main cave, a handful of possible related sites, and many places where Denisovan DNA appears in living people, but the archaeological deposits are still poorly known. It's entirely possible that Denisovans routinely used fire, hunted large game, processed hides and crafted tools at a level comparable to Neanderthals and early modern humans. Their genetic success across such varied environments strongly hints at that. You do not colonize the Tibetan plateau, survive Siberian winters, and reach islands in Southeast Asia without knowing how to control fire, manage resources, and teach complex skills to your children. Symbolic behavior is trickier. Did Denisovans string beads? Carve abstract marks? Tell stories around the fire, marking their memories with objects? Given how widespread symbolic behavior becomes later, in both Neanderthals and modern humans, and given that these groups all shared ancestry from the same bottlenecked population, many researchers suspect that at least a basic capacity for symbolic thought was present in their common ancestors. But capacity is not the same as expression. The archaeological record for Denisovans doesn't yet give us undisputed examples of art, or clearly deliberate symbolic markings that everyone agrees on. What we see instead are tantalizing hints, a possible bracelet here, a pierced tooth there, patterns of tool standardization that suggest teaching and tradition, but not yet the dense, decorated landscapes we find in later Homo sapiens sites. In the absence of clear signatures, archaeologists lean on method instead of romance. They compare Denisova Cave to better understood Neanderthal and modern human contexts. They ask, what would count as a robust association? What level of dating precision? What kind of stratigraphic clarity? What pattern of repetition across sites would we need before saying, with a straight face, this is Denisovan culture? We are not there yet. For now, Denisovan material culture sits in a twilight zone, partly visible, partly blended with that of their cousins and neighbours. We infer more from where they must have lived and what climates they survived than from objects we can confidently say they made. Future discoveries could change that almost overnight. A cave high on the plateau, with clear Denisovan DNA, well-preserved hearths, and undisturbed floors littered with the same distinctive ornaments, tools and perhaps even engravings. An open-air site in Southeast Asia, sealed under volcanic ash, with Denisovan genomes in the soil, and a coherent toolkit lying exactly where it fell. A waterlogged camp with preserved wooden implements, plant fibres, maybe even traces of pigments. Any one of those would begin to turn the smears into lines, the maybes into something closer to probably. Until then, Denisovans remain strangely lopsided in our knowledge. Their genes speak loudly. Their bones whisper. Their tools, fires, and possible symbols are more like echoes, heard faintly in the mixed chorus of other humans who shared their caves and their continents. We know they had culture because all humans do. We just don't yet know exactly what their particular way of being human looked like on the ground, in stone and ash and ornament. What we do know with absolute certainty is that some pieces of that culture did not vanish with their last campsites. Some of it is still here, right now, moving silently inside our cells. To see that side of the story, we have to leave the cave sediments behind and go back into the genome, where Denisovans are not shadows at all, but living threads woven into millions of modern bodies. If you could zoom into your own DNA and colour the Denisovan bits in glowing blue, what would you see? For most people listening to this right now, the answer is nothing at all, or almost nothing. A faint dusting of blue, a few scattered fragments buried under a mountain of other ancestry. But in some populations, especially across parts of Asia and Oceania, those blue threads are bright and unmistakable. Not because those people are somehow more Denisovan in any simple sense, but because in their distant past, their ancestors met Denisovans again and again, and some of what they exchanged stayed useful. The broad pattern looks something like this. People whose recent ancestry is mainly from Africa tend to have either no detectable Denisovan DNA or amounts so tiny they're close to the edge of current methods. That doesn't mean their ancestors never interacted with Denisovan-like groups somewhere, sometime. It just means that, within the limits of what we can measure right now, the Denisovan signal in most African genomes is very weak. As you move into East and Southeast Asia, a small but noticeable Denisovan component appears. On average, people from these regions carry more Denisovan ancestry than, say, people whose roots are mainly European or Middle Eastern, who tend to show little to none. Then the signal spikes. In many populations of island Southeast Asia and Oceania, especially in places like Papua New Guinea, some indigenous Australian groups, and certain Pacific Island communities, the estimated Denisovan ancestry can reach several percent. In genetics terms, that's enormous. It suggests not just one ancient encounter, but multiple episodes of contact and interbreeding with Denisovan-related groups over a very long time. The geographic pattern tells a story. It suggests that when early modern humans streamed out of Africa and fanned across Eurasia, they did not pass through a Denisovan-free world. In parts of Asia, especially further east and south, they kept running into Denisovan populations or their close relatives. Sometimes these meetings were fleeting. Sometimes they were intimate enough that children were born, and some of those children thrived. Over tens of thousands of years, those scattered unions left behind tiny genetic footprints. But here is where we have to be careful. When you hear numbers like 3% Denisovan ancestry, or 0.2%, or none at all, it's tempting to treat them as precise readings, like a fuel gauge in a car. They're not. These percentages are estimates. They come from statistical comparisons between modern genomes and the handful of Denisovan genomes we've reconstructed from tiny fragments of bone and tooth. Different research teams use slightly different reference genomes, different mathematical models, different assumptions about population history. So you will see the numbers move around, sometimes by a full percentage point or more between studies. One group might say, this population has around 4% Denisovan ancestry. Another might say, closer to 2 or 3%. Both are probably circling the same reality. There is a meaningful Denisovan component there, higher than in most other populations, but the exact number is fuzzy. Even the phrase Denisovan ancestry is a simplification. The Denisovans we know from Denisova Cave might not represent all Denisovan-like populations that ever live. There could have been multiple branches, some more divergent than others, some never sampled by our ancient DNA yet. When scientists say a piece of modern DNA looks Denisovan, what they usually mean is that it looks more similar to the Denisovan genomes we have than to Neanderthals or to the inferred ancestral modern human sequence. Think of it like hearing a familiar accent in a crowded room. You might not know the exact town the speaker is from, but you recognize the region. So how do geneticists actually find these Denisovan-like segments in our genomes? The basic idea is to line up DNA from lots of modern people, position by position, and compare it to ancient reference genomes. Most of your DNA will look like a shuffled mixture of very old human variants, spread and remixed over hundreds of thousands of years. But some stretches stand out. Every now and then, scientists find a segment where the modern sequence matches Denisova Cave DNA closely, is markedly different from Neanderthal and African sequences, and is also unusually long or structured in a way that suggests it entered the modern human population relatively recently in evolutionary terms. These are candidates for introgressed segments, pieces of DNA that entered the modern human gene pool from Denisovan ancestors through hybrid children, and then got passed down through the generations. It's a bit like reading a palimpsest manuscript, where a later text has been written over an earlier one. With the right tools and enough comparisons, the older writing begins to emerge. When you do this across thousands of genomes, patterns come into focus. Certain Denisovan-derived segments pop up again and again in particular regions or populations, and are rare or absent elsewhere. Some are short and scattered, echoes of very ancient mixing. Others are surprisingly intact, suggesting later interbreeding or strong natural selection preserving those variants. That brings us to one of the most important ideas in this whole story, adaptive introgression. Not all borrowed genes are equal. When modern humans and Denisovans had children together, those children would have received a random mix of genes from both sides. Over time, most of the Denisovan-derived variants would have drifted, diluted, and often disappeared, just as most random genetic variation does. But every once in a while, a Denisovan-derived gene did something especially helpful in a particular environment. Maybe it improved oxygen use at high altitude, or tweaked the immune system to better handle local pathogens, or adjusted how the body handled certain nutrients or temperatures. In those cases, natural selection could step in. Individuals carrying that helpful Denisovan variant would, on average, be slightly more likely to survive and have children in that environment. Over many generations, that particular chunk of Denisovan DNA could spread, even as most other Denisovan fragments faded into the background. This is adaptive introgression, not just mixing, but mixing that leaves behind very specific, functionally important pieces because they conferred an advantage. Sometimes those segments are so beneficial that they spread beyond the region where they first arose. In other cases, they remain localized, tightly tied to the landscape where they originally offered a survival boost. But before we dive into examples, there's a misconception we need to lay to rest. Having more Denisovan ancestry does not make anyone less evolved, more primitive, or less human. Those ideas come from a very old, very mistaken way of thinking about evolution, like a ladder with advanced humans at the top and archaic humans lower down. Real evolution does not work like that. It's a branching tree, a tangled web of lineages adapting to their own challenges. Denisovans were not unfinished versions of us. They were fully adapted humans for their time and place, shaped by the same brutal pressures of survival as our own ancestors. If anything, the genes we inherited from them that survived are likely to be the ones that worked especially well. When we talk about modern populations with higher Denisovan ancestry, we are not talking about people who are somehow closer to a bygone stage of humanity. We are talking about people whose ancestors picked up particular helpful tools from a neighbouring human group and kept them because they worked. Every person alive today has a mosaic genome, pieces from many ancient populations, most of them unnamed, most never to be found as bones in caves. Denisovan fragments are simply one recognisable colour in that mosaic, and they matter not because they mark anyone as different in some essential way, but because they show how deeply interconnected human history really is. Our species did not march out of Africa, wipe the slate clean, and start fresh. We wove ourselves into the existing human fabric, taking in threads from Neanderthals, Denisovans, and possibly other ghost lineages we have yet to pin down. Those threads now help us breathe, fight infection, metabolize food, and cope with cold and heat. In the next part of this story, we'll zoom in on some of those threads. We'll look at the Denisovan-derived variant that helps people live and work on the roof of the world, where the air is thin and the sky presses close. We'll explore immune system tweaks that may have come from ancient Asian Denisovans, shaping how some modern bodies respond to microbes. We'll touch on metabolic and physiological traits that whisper of long-gone environments. These are not just curiosities, they're reminders that evolution never stopped, that our bodies are still carrying the fingerprints of encounters that happened in dark valleys, mountain passes, and island shores tens of thousands of years ago. To follow those fingerprints, we turn from the broad picture of who has Denisovan DNA to the finer question. What exactly did those borrowed genes do, and how are they still working, quietly, inside us tonight? Let's start on the roof of the world. Imagine a young herder walking across the Tibetan plateau at dawn. The air up here is thin enough to make most lowlanders dizzy and sick within hours. Oxygen levels are about 40% lower than at sea level. For most human bodies, that's like living in a permanent state of suffocation. But for people whose ancestors have lived there for thousands of years, the body's response is different. Their lungs still work hard, their hearts still beat fast, but their blood doesn't turn into sludge. Most humans who move to high altitude respond by cranking up their red blood cell production. More red cells means more oxygen transport, but it also makes the blood thicker, harder to pump, and more likely to form clots. It's a short-term fix that doesn't age well. Many Tibetans and some neighbouring highland populations do something else. Their bodies keep red blood cell levels much lower than you'd expect at that altitude, closer to sea level values. They avoid the worst of the chronic mountain sickness that plagues newcomers. They can work, reproduce, and live full lives at heights that leave most of us gasping. Deep in their genomes, a gene called EPAS1 carries part of the explanation. EPAS1 is involved in sensing oxygen levels and regulating the body's response to low oxygen. In Tibetan populations, a particular version, a specific haplotype of this gene, is unusually common. When geneticists compared it to sequences from other humans, something strange happened. It didn't look like typical modern human variation. It didn't look like Neanderthal either. It looked Denisovan. Not identical, but close. Much closer than to the African or European sequences used as baselines. And it wasn't scattered randomly. The Denisovan-like version of EPAS1 in Tibetans sits in a long, intact block of DNA, exactly what you'd expect if it came in relatively recently from an archaic human and was then pushed hard by natural selection. In population genetics, this is known as a classic signature of adaptive introgression. You don't need much imagination to picture how it happened. Sometime after modern humans reached the highlands of Asia, they met local Denisovan-related groups, or descendants of those groups. Children were born with mixed ancestry. Most of the Denisovan DNA in those children drifted, recombined, and mostly faded over time. But not this bit. In the thin air of the plateau, the Denisovan-derived EPAS1 variant seems to have given carriers a small but real advantage. Maybe they were less prone to miscarriages at high altitude. Maybe their hearts and lungs coped better with long winters and hard labour. Whatever the exact mechanism, individuals with that variant had, on average, more surviving children. Generation after generation, that subtle edge added up. The EPAS1 variant spread through the population, fanning out until it became common among Tibetans. Today, when you look at their genomes, it stands out like a bright blue thread, a Denisovan-like island in a sea of mostly modern human ancestry. How strong is the evidence for this story? Among the various Denisovan adaptation tales, this is one of the best supported. The EPAS1 signal in Tibetans has been replicated by multiple independent studies. The sequence similarity to Denisovan DNA is unusually high. The statistical signatures of strong recent selection around that gene are robust. Most researchers who work on this agree on the broad outline. A Denisovan-like variant of EPAS1 entered the modern human gene pool and was favoured in high-altitude populations. But even here, with one of our clearest examples, the details remain tricky. We don't know exactly which population of Denisovans contributed it. The Denisovan genome from the Siberian cave is our reference, but the real donor might have been a related group further south or east, slightly different, but close enough to show up as Denisovan-like. We also don't fully understand the trade-offs. Genes that are helpful in one environment can be neutral or even harmful in another. The Denisovan-derived EPAS1 variant might have subtle costs at low altitude or in different disease landscapes. Those effects could be too small to measure easily, but evolution is always balancing such trade-offs. Still, as far as adaptation stories go, high-altitude EPAS1 is about as real as it gets. A sharp, specific case where an archaic gene appears to have been pulled out of the genetic rubble and polished by natural selection until it shone. Altitude isn't the only place Denisovan DNA seems to matter. When modern humans moved into Eurasia, they weren't just dealing with thinner air or colder winters. They were stepping into new disease ecologies, unfamiliar parasites, viruses, bacteria, fungi. Pathogens that had been co-evolving for hundreds of thousands of years with the local animal communities, and with local human groups, including Neanderthals and Denisovans. Our immune system is not one thing, it's a whole orchestra of genes and pathways. HLA genes that help the body recognize foreign proteins, toll-like receptors that detect microbial patterns, cytokine signaling networks that turn inflammation up or down. Across parts of Asia, some of these immune-related regions carry Denisovan-like segments that appear more common than you would expect by chance. In some cases, the patterns look suspiciously like adaptive introgression. For example, certain toll-like receptor variants that help detect bacterial and fungal invaders show archaic signatures. Some look more Neanderthal-like. Others tilt toward Denisovan-like sequences, especially in Asian populations. These variants can influence how strongly the body responds to infections, and possibly how it balances the risks of underreacting versus overreacting. There are also Denisovan-like contributions in other immune regions, pieces that may tweak antiviral defenses or shape how inflammation is regulated. The logic is similar to the altitude story, but messier. A modern human group entering a new region might have been vulnerable to local pathogens that Neanderthals or Denisovans had been wrestling with for millennia. Hybrid children who inherited a particularly useful immune variant from a Denisovan parent might have fared slightly better. That tiny fitness advantage could, over time, cause that variant to spread, even if most of the other Denisovan DNA in the population vanished. We can see the outlines of this in the data. Clusters of Denisovan-derived alleles in immune genes, higher frequencies in regions where contact with Denisovan-related groups likely happened, signs of non-random survival. But here, the evidence is less clear-cut than with EPAS1. Immune genes are highly variable and subject to many different pressures. Pathogens change, cultural practices shift, population sizes expand and contract. It's harder to tease apart, this is here because it came from Denisovans and was strongly selected, from this is here because it drifted, recombined, and happened to stick around. Some studies argue strongly for adaptive introgression at particular immune loci with Denisovan-like sequences. Others caution that demographic history and random processes can mimic some of the same signals. The field is very much in motion. This is where it helps to step back and remember how natural selection works on introgressed DNA. When two human groups mix, the child inherits entire chromosomes from each parent, massive blocks of DNA. At first, those blocks are long and cleanly separated. Then, generation after generation, recombination slices them into smaller pieces. Each child gets a shuffled deck, half from one parent, half from the other, but each parental half already a mosaic of older ancestors. If there is no particular advantage or disadvantage to most of the archaic DNA, it simply fragments and drifts. Over tens of thousands of years, only faint, scattered crumbs remain. Short stretches of Denisovan-like sequence here and there, often so subtle they're almost indistinguishable from ordinary human variation. But if a particular archaic segment is helpful in a given environment, selection acts like a magnet pulling that segment upward in frequency. People carrying it leave more descendants on average. Those descendants carry the same segment, along with a bit of surrounding DNA. Recombination keeps chopping it at the edges, but the core piece under selection can remain intact for surprisingly long periods. At the population level, you see a distinctive pattern, a chunk of archaic-like DNA that is both unusually common and unusually long. That's what we see with Tibetan EPAS1. That's what we may be seeing around some immune genes in Asia and Oceania. A few pieces of Denisovan code that refuse to fade because the environment kept rewarding them. But this is also where we have to be very careful. It is tempting to tell simple, satisfying stories. This Denisovan gene gives better immunity. That one makes you stronger in the cold. This one explains an entire population's health profile. Reality is rarely that neat. Most traits we care about, disease resistance, altitude tolerance, body shape, metabolism, are influenced by many genes, each contributing a tiny effect, and by layers of environment and culture on top of that. Diet, childhood infections, social organization, even technology, all interact with biology in complex ways. When scientists say a particular Denisovan-derived variant is associated with a trait, it usually means that in statistical studies across many people, those who carry the variant show a slightly different average value for some measured feature. That doesn't mean the variant single-handedly causes the trait, or that everyone with it will experience the same outcome. It also doesn't mean we fully understand what the variant is doing. Sometimes we find a Denisovan-like segment near a gene with a plausible function, like an immune receptor, and we infer a role. Other times the functional data is thin, or comes from laboratory systems that only approximate the complexity of a living person. So when you hear that Denisovan ancestry gave Tibetans their high-altitude abilities, or armed Asians with superior immunity, it's worth mentally softening those verbs. A better way to say it might be, Denisovan introgression contributed a few specific genetic variants that in certain contexts were folded into the ongoing evolutionary experiments of modern human populations. Some of those variants seem to have helped. Many probably did nothing obvious. A few might even have been harmful in some settings and advantageous in others. The Ice Age world these populations inhabited sharpened every edge. Cold, drought, famine, and disease kept grinding away at the edges of human survival. In such a world, even small advantages, a slightly better oxygen response at altitude, a slightly more tuned-in immune receptor to a local pathogen, could spell the difference between a family line that continues and one that ends. Denisovans, Neanderthals, and modern humans were all subjected to these pressures. They all evolved their own local solutions: body shapes, metabolic quirks, immune tweaks. The great mixing events, those periods when their descendants had children together, didn't just blur separate stories into one. They allowed those solutions to be shared, tested, and sometimes adopted. From the quiet perspective of your body lying in the dark, listening now, it means something simple and profound. Inside you, there are genetic fragments that survived multiple rounds of natural selection. First in archaic humans weathering Ice Age extremes, then in the hybrid populations that inherited their genes, and finally in the long chains of ancestors that led to you. Some of those fragments may have started life in a Denisovan child staring out over a glacier, or a Denisovan healer experimenting with strange new plants in an Asian forest. We can identify a few of those fragments with some confidence. Epus 1 in Tibetans, a handful of immune variants in parts of Asia and Oceania. Others are still shadows in the data, debated and reinterpreted as new methods come online. Many more are probably hiding just below the threshold of what we can currently see. As we move forward, it's worth holding both sides of this story at once. On one hand, adaptive introgression from Denisovans is real. It has left measurable functional marks in some modern populations. It shows that human evolution was not just about competition, but about borrowing and sharing. On the other hand, we are only beginning to understand what those borrowed genes actually do. Each claim needs careful testing. Each elegant story about a Denisovan gene for X has to be checked against messy data and alternative explanations. In the next part of this journey, we'll step back even further. We'll look at how discoveries like these are forcing scientists to redraw the human family tree, not as a neat set of branches, but as a tangle of overlapping lineages, ghost populations and genetic bridges. We'll ask what it really means to say that Denisovans weren't what we were told, and how their legacy, and the legacy of other lost humans, changes the way we think about ourselves. For a long time, the way this story was told was comfortingly simple. You may have heard some version of it in school, or in a documentary with clean diagrams and tidy arrows. First, there were primitive ape-like ancestors, then Homo erectus, then maybe Heidelbergensis, then Neanderthals in Europe, archaic humans in Africa, Denisovans in Asia, and finally us, one species after another, replacing the previous one like new models of a phone, a straight line with a few side branches that ended in dead ends. Neanderthals were usually cast as the classic dead end. Denisovans, when they were added to the story, slotted naturally into the same role. A mysterious cousin who appeared, did something, then vanished when we arrived. It's an appealing picture, clear, linear, easy to remember. It treats species like discrete boxes with firm edges and neat start and end dates. It suggests there was a moment when modern humans emerged, fully themselves, and everything else was swept aside. The trouble is, the more data we collect, the less that picture matches reality. The genetic evidence from Neanderthals was the first crack. When their genome was sequenced, it didn't sit cleanly outside of us. Pieces of it were inside us. Non-African populations carried a few % of Neanderthal ancestry. That meant interbreeding. That meant overlap. That meant Neanderthals were not just replaced. They were, in a small but real way, absorbed. Denisovans deepened the crack into a fracture. Their DNA isn't just a small smudge in a few people. In parts of Melanesia and Australasia, Denisovan-like ancestry can reach 5% or more. In Asia, distinct pulses of Denisovan-related introgression show up at different times and places. Not one contact event, but several. Not one uniform Denisovan population, but multiple deeply diverged lineages, some closer to the Siberian cave genome, others far enough away that they may have split hundreds of thousands of years earlier. When geneticists try to map these signals onto a family tree, the tree stops looking like a tree. It starts to look more like braided rivers. Instead of a single trunk splitting cleanly into separate branches, you see streams dividing, rejoining, and exchanging water as they move across the landscape. Populations separate for a while, adapt to different local conditions, and then meet again. Children are born with ancestry from both sides. Genes flow across the boundary. Then the groups may drift apart again, carrying bits of each other into the future. This is the newer view taking shape in human evolution. Not a replacement model, but a reticulate one. Not tidy speciation events that slam doors shut, but long periods of partial separation with occasional doors swinging open, letting genes slip back and forth. In this braided stream picture, modern humans, Neanderthals, and Denisovans are not three perfectly isolated species. They are three major lineages within a wider interlinked network of Pleistocene humans, all of them descendants of that ancient bottleneck, all of them still close enough genetically that when they met, at least some of the time, they could have fertile children. That doesn't mean they were all one big panmictic population. It doesn't mean species boundaries are meaningless. It means the boundaries were porous, semi-permeable membranes rather than brick walls. Denisovans sit right on that conceptual fault line. From a textbook perspective, you might ask, were they really a separate species in the same sense that, say, humans and chimpanzees are separate species? Many paleoanthropologists still refer to them as Homo denisova or a subspecies of Homo sapiens, because they are clearly distinct from both Neanderthals and us in genome-wide comparisons. They form their own clade, their own branch. But the level of difference is modest compared to many other species pairs, and the existence of fertile hybrids like Denny, the Neanderthal Denisovan daughter, tells us that whatever we call them, reproductive isolation was not complete. The same is true for Neanderthals and modern humans. Genetically, we were diverged enough to be distinguishable, but not diverged enough to be entirely separate in practice. So what does species even mean here? Biologists have a notoriously hard time answering that question in general, and Pleistocene humans make it harder. There is the biological species concept, which focuses on interbreeding. There are morphological concepts, which focus on anatomy. There are genetic concepts, which look at divergence times and patterns of gene flow. Denisovans complicate all of them. Their bones are too fragmentary to give us a full anatomical picture. Their genetic divergence from Neanderthals and modern humans is real, but not vast. Their ability to produce hybrids is demonstrated. Their genetic legacy persists in living people. If you draw the line tightly, insisting that a species must be completely reproductively isolated, Denisovans, Neanderthals, and modern humans start to look more like divergent regional populations of one very flexible, wide-ranged human metaspecies. If you draw the line more loosely, focusing instead on long-term genetic and ecological distinctness, they comfortably qualify as separate species that happen to have limited hybrid zones where they overlapped. Most researchers land somewhere in the middle. They keep using species names because they're useful shorthand for major lineages with different histories and adaptations, but they accept that those species interacted and mixed in ways that don't fit older, cleaner stories. This is part of why early media coverage of Denisovans went the way it did. When the first papers came out, the headline, "New Species of Human Discovered," was irresistible. It frames the find as a clear addition to a pre-existing list. Neanderthal, check. Denisovan, check. Another box filled in on the chart. The press likes singular things with crisp labels, the Denisovans, as if there was one coherent group, one culture, one look, one fate. The reality, as we've seen, is fuzzier. Those Denisovan-like signals in modern genomes split into at least two, and probably more, clusters. The DNA from the Siberian cave represents one lineage, likely living in colder northern environments. Other Denisovan-related sequences in island Southeast Asia seem to trace back to a much older, more southern population. These lineages were as different from each other as Neanderthals were from us, maybe more. Yet they all get flattened into a single category, Denisovan. Part of that flattening is practical. We only have one high-quality Denisovan genome so far, from that cave. It becomes our reference, our label. Part of it is cognitive. Our brains like stories with a manageable cast of characters. Modern humans, Neanderthals, Denisovans, feels tellable. Multiple deeply diverged Denisovan-related lineages that interacted in complex ways with multiple waves of modern humans does not fit easily into a 2-minute news segment. So the older narrative persisted. Neat branches, sequential replacements, a lone mysterious cousin vanishing from the stage. But as you lie here listening, you can let that picture dissolve a bit. Imagine instead that for hundreds of thousands of years there were many kinds of humans sharing the planet. Some looked more like us, some less. Some were robust, cold-adapted hunters in Europe. Some were high-altitude specialists on the Tibetan plateau. Some navigated island chains. Others moved through African savannas and forests. Their ranges overlapped and shifted as climates changed. Boundaries between them were sometimes sharp, sometimes blurry. At the genetic level, this produced a mosaic. Each of us today carries small pieces from different ancestral tiles, a primarily African modern human base layer, dusted here and there with Neanderthal segments, speckled with Denisovan-like fragments in some populations, and potentially touched by contributions from other yet unseen groups. No single person is a Denisovan hybrid in the simple sense. Instead, human populations are long-running experiments in mixing and remixing, with local environments and histories tilting which fragments persist. This mosaic view doesn't erase difference. It doesn't say everyone was the same. It does something subtler. It says that difference and connection coexisted. Lines diverged, but they never stopped talking to each other entirely. In the mainstream scientific community, this braided mosaic ancestry picture has been gaining ground for years. It's supported by genomes from Neanderthals, from Denisovans, from ancient modern humans scattered across Eurasia and Africa. It's reflected in models that allow for multiple pulses of gene flow, not just one out-of-Africa event, sweeping the board clean. It dovetails with fossil evidence that shows overlapping forms and gradual transitions, rather than sudden global turnovers. At the same time, there are still open questions. How many distinct Denisovan-related lineages were there? How far back did they diverge from each other? How frequent was interbreeding, and was it symmetrical or mostly one-sided? Did some regions of the genome remain largely resistant to mixing, preserving deeper differences beneath the mosaic surface? We have hypotheses, simulations, partial answers, but not a final map. What does seem clear is the lesson. Our ancestry is not a single line of descent from a small, pure group that outcompeted all rivals. It's a braided network of populations moving, splitting, meeting again, sometimes fighting, sometimes trading, sometimes having children together. It is shaped by climate swings and chance encounters, by glacial advances and retreats, by rivers shifting course and coastlines rising and falling. Denisovans are important not just because a few of their genes help some people handle altitude or disease. They matter because they force us to loosen our grip on simple stories about who counts as us and who counts as them. They remind us that human has always been a moving target, a cluster of overlapping possibilities rather than a single static form. They show that our species, if we even want to use that word in the strict sense, was born not in isolation, but in contact zones, places where different kinds of humans met, mixed, and reshaped each other. Somewhere in that tangled history, a child with one Neanderthal parent and one Denisovan parent, lived out her brief life in a Siberian cave. Somewhere else, a child with a Denisovan-derived oxygen gene took her first untroubled breaths on a high plateau. Their stories did not end with them. Pieces of those stories moved forward. They're still moving now, in the quiet cells of your body, in the patterns of your blood and bones and immune responses. They came from a past that was nothing like the simple chart on a classroom wall. In the next section, we'll follow that tangle even further into the realm of ghost ancestors, groups we can detect only through their genetic echoes, with no bones at all to hold in our hands. If Denisovans complicated the family tree, those ghosts threaten to redraw it entirely. When geneticists talk about ghost populations, they mean something very simple and very unsettling. A ghost population is a group of ancient humans we can see in the DNA, but not in the ground. There are no skulls in museum drawers, no jawbones with labels, no carefully dated skeletons. Instead, there are strange patterns in genomes, segments that don't match Neanderthals, don't match Denisovans as we currently define them, and don't match any known modern human ancestors. They are too different to be explained as ordinary variation, and too coherent to be random noise. Somewhere, sometime, there must have been a population that carried those sequences. They lived, they had children, they met other humans. They left no recognized fossils behind, but they left marks in the double helix of people who came after. Those are the ghosts. You've already met one version of this idea without quite naming it. When we say that Denisovan-like ancestry in island Southeast Asia looks extremely old and deeply diverged from the Siberian Denisova Cave genome, we are really saying something Denisovan-ish was there long before the cave population, maybe long enough to count as a separate branch. We call it Denisovan because we don't have a better word yet, but the DNA suggests a more crowded stage. In Africa, where our lineage spent most of its history, multiple studies have found signals of archaic introgression that don't fit Neanderthals, don't fit Denisovans, and can't be matched to any known fossil group with sequenced DNA. Some West and Central African populations carry tiny fragments that seem to have split off from the rest of the human family tree hundreds of thousands of years before our own line. The simplest explanation is that there were other kinds of humans in Africa, archaic groups that overlapped with early Homo sapiens and exchanged genes with them. We don't know what they looked like, we don't have their tools. We don't know what they called themselves. We only know that pieces of their genome still circulate in people living today. Outside Africa, the picture is just as eerie. In Asia, certain genetic patterns don't sit comfortably on a simple three-way model of modern human Neanderthal Denisovan. Some sequences in modern East and Southeast Asian populations look like they came from an archaic source related to Denisovans, but not especially close to the Siberian sample. Others look like they might trace back to a lineage that separated before Neanderthals and Denisovans themselves diverged. Again, the neat labels fray. One way to think about this is as background noise. Maybe the models are complicated. Maybe subtle biases are tricking us. And sometimes that's true. Statistical methods can hallucinate ghosts where none exist, especially when the signals are faint and the assumptions are off. But the same kinds of signals keep turning up, in study after study, using different datasets and approaches. They don't all agree on the details. They do suggest that Denisovans are not the only missing branch. So what might these unseen humans have been? One possibility is that some of them are already sitting in our fossil record, misfiled under broader labels like archaic Homo or enigmatic specimen, a skullcap in a drawer, a jawbone from a cave, a partial skeleton that doesn't quite fit Neanderthals or modern humans, but hasn't yet been linked to a genome. Until we extract DNA from those bones, or proteins that can be used as genetic proxies, we can't match the skeletons to the ghosts in our models. Another possibility is that some of them have not been found at all. If you look at a map of places with rich Pleistocene human fossils, you'll notice clusters. Europe, parts of the Levant, a smattering of sites in China and Indonesia, some caves in South Africa. Then you notice the blanks, vast regions of forested Central Africa, much of India, large swaths of mainland Southeast Asia, the interiors of Borneo, Sulawesi, New Guinea, areas where climate and geology make fossil preservation difficult, and where modern development, thick vegetation, and limited funding make surveys hard. Yet these are precisely the regions where genetic signals hint at complex histories. Island Southeast Asia, in particular, keeps drawing attention. It's where we find Homo floresiensis, the tiny hobbits of Flores. It's where Homo luzonensis turned up in a Philippine cave, with its odd combination of primitive and derived traits. It's where Denisovan-like ancestry in modern populations appears both strong and unusually ancient. And it's a region where moving between islands has always required some level of seafaring, even in periods of low sea level. If Denisovan-related groups were here for hundreds of thousands of years, there was room for them to fragment, adapt locally, and diverge into multiple distinct lineages. Some might have specialized in montane forests, others in coastal zones, others in river valleys or limestone karst. Some might have remained small and isolated enough that from a genetic standpoint they became as different from each other as Neanderthals were from us. Today all of that diversity is collapsed into a single label, Denisovan, but that label is a placeholder, not a final verdict. As more data come in, it is entirely possible that what we currently call Denisovans will split the way reptiles once did in biology textbooks. A convenient category will be broken into multiple, more precise branches. We may end up speaking of several named species or subspecies, a northern Denisovan lineage associated with Siberia and the Tibetan Plateau, one or more southern lineages in island Southeast Asia, perhaps another on the Asian mainland. All related, all sharing a deeper ancestor, but distinct enough in their own right to warrant separate names. What would it take to justify that kind of splitting? We would need converging lines of evidence, not just ambiguous genetic hints, but ancient genomes or proteomes from actual bones that show clear groupings, fossils with anatomical traits that track those genetic differences, radiometric dates that place those groups in different times and environments with limited overlap. At the moment we have only part of that picture. We have excellent genomes from a small number of Neanderthals. We have one high-quality Denisovan genome and some lower coverage ones. We have a scattering of ancient modern human genomes. We have many more fossils with no DNA at all. When researchers build models to explain the patterns seen in living people, they are doing something like reconstructing a symphony from a handful of surviving notes. Alternative interpretations are always on the table. It could be that some of the ghost signals we see in Africa reflect deep population structure within early Homo sapiens themselves, rather than gene flow from a truly separate species. In other words, our own ancestors might have been subdivided into long separated groups that occasionally mixed, producing patterns that look from a distance like archaic introgression. In Asia, some signals might be explained by unsampled Denisovan diversity that doesn't require an entirely new category, just more variation within the broad Denisovan umbrella. Or by ancient population splits and bottlenecks among early modern humans, leaving echoes that resemble archaic admixture. How do we tell the difference? Different scenarios leave different fingerprints. Introgression from a genuinely archaic group tends to create distinct blocks of DNA with high divergence from everything else, often with particular shapes in the frequency spectrum of variance. Deep structure within a single species can mimic some of these features, but usually not all of them at once. By comparing thousands of regions across the genome, and by testing many possible histories in simulations, researchers can start to discriminate between ghost ancestor models and complicated but still within one species stories. Ancient DNA is the most decisive test of all. When we sequence a bone and discover that its genome matches one of the hypothesized ghost lineages, the ghost becomes a person, or at least a population. When instead a bone's DNA slots into an already known branch, some ghost models shrink or vanish. This is why every new Pleistocene genome feels like opening another window in a dark house. In the coming decade, those windows are likely to multiply. Ancient DNA techniques keep improving. Proteomic methods, reading the sequences of ancient proteins when DNA is gone, are extending our reach back in time and into warmer regions where DNA decays quickly. More fieldwork is happening in previously neglected areas. Laos, Sulawesi, the Philippines, the interior of Papua, understudied parts of India and China, caves in tropical Africa. Each new site, each new fragment, is a chance to redraw the map. We may find that the Denisovan label fractures into a small constellation of related forms. We may finally connect the ghost signals in African genomes to specific fossils, giving faces, however partial, to those unnamed ancestors. We may discover entirely unexpected lineages that don't fit any current model, forcing us to widen the frame yet again. Or we may learn that some of the wilder ghost stories were just artifacts of limited data, and that the real picture, while still complex, is somewhat simpler than our first, overfitted reconstructions. Either way, the direction of travel is the same. The blank spaces on the chart are filling in, not with a single replacement wave sweeping clean, but with overlapping histories of contact and coexistence. Denisovans were our warning that the story was richer than we'd been told. The ghost populations are the whisper that even that richer story is incomplete. As you drift with these thoughts, you are living in a brief pause between discoveries. Somewhere right now, in a cave whose entrance is choked with vines, or under the floor of a rock shelter in Borneo, or in a riverbank in West Africa, there are bones lying in the dark, waiting. Bones from people who were not quite us, not quite Neanderthal, not quite Denisovan as we currently imagine them. People who met our ancestors, shared genes with them, and then disappeared. Their genomes are still here, moving quietly through the generations, stitched into the fabric of living bodies. In the next section, we'll turn from those hidden ancestors to the question of what they mean for us now, not as data points, but as part of how we think about identity, belonging, and the strange, enduring presence of lost humans inside the living. When you hear that you carry Denisovan DNA, or Neanderthal DNA, or traces of some unnamed ghost ancestor, it can feel strangely personal. These aren't just abstract percentages on a lab report. They're threads woven into your blood, your bones, the way your lungs handle thin air, or your immune system meets a new infection. They are echoes of specific encounters. A child born from a meeting on a windswept plateau, or in a forest valley, or beside some ancient shoreline that no longer exists. But what does that actually mean for who you are? One temptation is to treat these discoveries like a new kind of horoscope. To say, I have this much Denisovan, therefore I am more X or less Y. Stronger, hardier, more ancient, more authentic. Another temptation is the reverse, to treat archaic ancestry as some sort of stain, something to minimize or wash out of the story. Both instincts miss the point. From the perspective of population genetics, what Denisovan ancestry does is help explain why bodies differ in small, practical ways, who copes more easily with high altitude, who has particular immune responses, who processes certain nutrients efficiently. These are adaptations, not verdicts. They say nothing about the worth of a person, the value of a culture, or the depth of anyone's humanity. There is no genetic ladder with Denisovans, Neanderthals, and modern humans stacked neatly from primitive to advanced. Evolution doesn't work like a staircase. It works like a branching thicket. Each branch is shaped by its own problems and its own solutions. Denisovans were not less evolved than us. In their time and place, they were doing exactly what we are doing now, trying to keep children alive, trying to make sense of the world, trying to hold communities together. The genes that survive from them are there because, in certain environments, they helped. High-altitude variants passed from Denisovans to the ancestors of modern Tibetans, Sherpa and related groups are a clear example. Those tiny changes in how hemoglobin and related systems handle oxygen made life at 4,000 meters a bit more sustainable. They didn't make anyone morally better or worse. They just made it slightly easier to carry water, herd animals and have healthy pregnancies where the air is thin. The same is true for immune genes. If you live in an environment with particular pathogens, an archaic variant that happens to recognize those microbes can be a quiet advantage. It might mean fewer fevers, fewer miscarriages, fewer funerals. Over generations, that advantage leaves a mark in the genome. A little cluster of archaic alleles at higher frequency than chance would expect. Again, these are stories about survival, not status. So when we talk about Denisovans living on inside us, we have to be careful with our language. It's easy, without meaning to slip into old hierarchies, to label some ancestries as purer, some as mixed, some as ancient in a way that's supposed to confer special authority or special burden. But if there is one clear lesson from this whole story, it's that everyone is mixed. Neanderthal, Denisovan, ghost populations in Africa and Asia, deep structure within early Homo sapiens themselves. Once you zoom out to the scale of hundreds of thousands of years, the idea of a single, clean, isolated lineage starts to look like a myth. Human history is a constant pattern of splitting and meeting again, diverging and reconnecting, braiding and unbraiding. Population history is not the same thing as personal identity. Your genome is a record of what happened to countless strangers you will never know. People who lived and died long before your language existed, long before your religion, long before your family name. They are part of the story that made your body possible, but they are not the same as yourself. Identity is what you build in the present out of memories, communities, choices, and commitments. You can honour the fact that your ancestors once met Denisovans or Neanderthals or some unnamed archaic group without turning that fact into a ranking system. You can be curious about the deep past without letting it be used as a weapon in arguments about who belongs where, because belonging is always local and always lived. If you stand today in a highland village in Papua, or a Tibetan monastery roofed in prayer flags, or a coastal town in the Philippines, the Denisovan story looks different each time. In Papua and parts of Melanesia, a relatively high fraction of Denisovan-related ancestry is present. In Tibet, certain Denisovan-derived variants shape altitude tolerance. In Southeast Asia, traces of multiple Denisovan-like lineages seem to flicker in the background of the genome. For a long time, those communities were treated in global narratives as marginal, at the periphery of big history. Now Denisovan research is quietly rewriting that map. Suddenly places like the New Guinea highlands or the Philippine archipelago are not footnotes. They are central to understanding how many kinds of humans once lived, and how our lineages intertwined. The same is true for parts of mainland Asia, the Tibetan Plateau, the forests and river systems of southern China, the caves of Laos and Vietnam. Denisova DNA has turned them into key chapters in a shared human chronicle. It's as if lines that once seemed separate are now revealed to be connected. A grandmother in the Himalayas, a farmer in coastal China, a fisher in island Southeast Asia, a child in urban Australia. All linked by tiny signals that point back to ancient meetings on landscapes we will never see. When you think this way, national borders and modern ethnic categories start to feel very recent, very thin. Deep time smooths the sharp edges of those divisions. 1,000 years ago, your ancestors may have been on different sides of a war. 50,000 years ago, they may have shared a shelter, traded tools, or exchanged a song neither side could now recognize. 200,000 years ago, they were all simply part of a scattered, fragile set of populations trying not to vanish in a changing world. Remembering that can be calming. Instead of imagining yourself as perched alone on the edge of history, you can imagine yourself as lying in the middle of a vast, woven blanket of ancestry. Some threads are bright and recent, the grandmother you knew, the language you speak, the stories you heard as a child. Others are faded and buried. Denisovan adaptations, Neanderthal bone density genes, ghost segments from nameless African or Asian lineages. But they all share the same loom, the earth, its climates, its dangers, its brief windows of abundance. This is one reason stories like the Denisovans can feel oddly soothing at night. They pull your attention away from the immediate noise, deadlines, headlines, the small anxieties that circle the mind, and let you drift over a much longer timescale. On that scale, arguments about purity, about who was here first, about whose ancestors are more original, all start to look strangely small. Every culture alive today is both ancient and recent. Ancient, because its roots sink into those same Pleistocene struggles, those same experiments in cooperation and meaning-making. Recent, because the specific words, customs and political boundaries we recognize now are the products of the last few thousand years at most, a blink compared to the 500,000 years that separate us from the first Denisovan ancestors. Somewhere in that awareness, there is room to relax. To think, my body is a temporary meeting of long stories. My thoughts tonight are a brief flare on the surface of something that has been going on, quietly, for a very long time. I am not separate from that. I am an instance of it. If you'd like to explore this whole saga in more depth when you're not half asleep, the full book version of this story is available at sleepytimehistory.com, and there's a small discount running this week. But for now, you don't need to remember links or dates. You can just let the idea of deep time settle around you like a blanket, because the legacy of Denisovans and their cousins is not locked in museum drawers. It's in every breath you take, in the way your cells handle oxygen and sugar and sunlight. It's in the quiet fact that you come from a lineage that has faced repeated disasters, bottlenecks, and near misses, and is still here. In the final part of this journey, we'll bring those threads together. We'll look directly at the notion that lost humans are still living inside us, not as ghosts to be feared or trophies to be claimed, but as reminders of how precarious, resilient, and interconnected our story has always been. And we'll ask what it means in a practical, everyday sense to carry that history into tomorrow. When you zoom all the way out, past the individual genes and the specific cave sites and the arguments between specialists, the Denisovan story rests on something surprisingly small. A handful of bones and teeth, a finger bone from a child in Siberia, a molar the size of a pebble, a fragment of skull, some tiny mineralized traces from caves in Tibet and Southeast Asia. If that were all we had, Denisovans would be nothing more than a footnote, an odd archaic population tucked into a single valley, living and dying unseen. But that is not all we have. From those fragments, scientists pulled DNA. From that DNA, a whole hidden history started to unfold. A separate branch of humans as distinct from Neanderthals as Neanderthals are from us. A population that split off early and then flourished quietly for hundreds of thousands of years. A lineage that met and mixed repeatedly with our own. We now see their traces not just in one cave, but in living people. A few % of the genome in parts of Papua and Melanesia. Particular altitude genes on the Tibetan plateau. Ghostly echoes scattered across East and Southeast Asia, hinting at multiple Denisovan-like populations we haven't even named. The map of human history that once looked like a clean line, Africa to Eurasia to the world, now looks more like a tangled river delta, with Denisovans running through many of its channels. The core evidence is modest: a few fossils, powerful genetics, and careful comparison. But put them together, and the picture they paint is startling. Denisovans were almost certainly not a single isolated group. They were likely diverse, with different branches adapted to different environments, from Siberian forests to tropical islands and high Himalayan plateaus. They seem to have been widespread, extending from the cold north to the steaming south, and deeply intertwined with us and with Neanderthals. We did not pass them on the road once and move on. We lived alongside them. We had children with them. We exchanged genes that still shape how some of us breathe, fight infections, and use energy. In that sense, they never really disappeared. The bones in the cave are gone. The people who lived there are gone. But the solutions they discovered to the problems of their world are still quietly at work, inside other bodies on other landscapes, inside ours. At the same time, there's a lot we still don't know, and it's important to keep those unknowns in view. We don't yet have a complete Denisovan skeleton. We can model their faces from genetic clues and comparisons, but we've never stood in front of a full Denisovan frame the way we have with Neanderthals. We don't know the full range of their cultures, their tools, their art or music, if they had them. We have hints of advanced technologies, of seafaring, of plant medicine, but hints are not the same as a finished story. We don't yet know how many Denisovan populations there were. The genetics suggest at least 3 major branches, possibly more, including lineages that split from each other hundreds of thousands of years before they met our ancestors. That means when we say Denisovan, we may be lumping together groups as different from each other as we are from Neanderthals. Somewhere in that diversity, there were likely distinct languages, distinct customs, distinct ways of making sense of the night sky and the animal world. We also don't fully understand how they vanished. Some Denisovan lineages may have faded gradually, their populations absorbed into expanding modern humans, their identities dissolving without a single decisive event. Others may have dwindled as climates shifted yet again, or as key food sources disappeared. Still others might have survived longer than we think, leaving traces we haven't yet recognized because we haven't looked in the right places, or because their bones have not yet reappeared from the earth. New discoveries could change this picture quickly. Another cave, another tooth, another high-quality genome from a region we hadn't sampled before. Each of these could redraw the map again, showing new branches, new meetings, new exchanges. A site with clear Denisovan tools and hearths might finally let us connect their DNA to a specific way of living on the land. A burial could show us their rituals, their relationship to death, their ideas of meaning. Even the genes we already know about are not fully understood. Some Denisovan variants in living people affect immunity, metabolism, or brain development in ways we are only beginning to trace. Future research may reveal that certain medical conditions, certain disease resistances, even certain sleep patterns are shaped in part by archaic ancestry. If that happens, we'll have to refine our stories again, being careful, once more, not to turn adaptation into hierarchy. All of this uncertainty can be strangely comforting. It means the past is not a closed book we must memorize correctly. It is an unfolding story, and we are alive at the moment when forgotten chapters are being found and stitched back in. There is room always for a new cave, a new insight, a new way of connecting dots that once seemed scattered. If you're listening to this as you fall asleep, you don't have to hold on to every detail. You can just let the broad outline sink in. You are the descendant of a line that passed through bottlenecks and expansions, droughts and ice, meetings and partings. Along the way, your ancestors met other humans who looked a bit different but were close enough to love, close enough to raise children with. Those encounters left quiet marks in your cells. In that deep sense, there are lost humans still living inside you. Not as complete people with names and faces and memories, but as patterns, solutions to old problems carried forward because they helped someone survive long enough to have a child, who had a child, who had a child, until the line reached you. Denisovan altitude genes in a highland shepherd. Neanderthal immune genes in someone walking through a modern city. Ghost DNA from Africa in a person who has never left the continent their ancestors flourished on. Across the globe in every time zone, people are lying down right now with those same long stories folded into their chromosomes. Someone listening from the islands of Southeast Asia where Denisovan traces flicker. Someone in the mountains where their ancestors learned to live in thin air. Someone in a lowland city, far from caves and glaciers, whose genome still carries whispers of cold plains and forest edges. Modern borders and identities are many and complex, but beneath them runs a shared, braided ancestry that does not stop at lines on a map. You are part of that. As you rest, you might imagine the world in darkness, turning slowly, little clusters of light where people are listening from. A village, a town, an apartment in a high-rise, a ship at sea, each one containing bodies built from the same ancient toolkit, adjusted in slightly different ways, all of them descendants of populations that once looked out at an unfamiliar horizon and decided somehow to keep going. If you've been drifting with this story and you'd like more journeys like it, you're very welcome to gently tap like or subscribe so you can find your way back here the next time you want to fall asleep to deep time. If something in the Denisovan story resonated with you, you can leave a quiet comment when you're awake. Maybe tell us where you're listening from. Which corner of this shared planet is home for you tonight? And if you find yourself wanting to go deeper into these prehistoric secrets when you're more awake, there's also a book that expands this whole saga. More slow walks through bottlenecks, ice ages, lost cousins, and the long road to now. You can learn more about it and pick up a copy if you like at sleepytimehistory.com. You don't need to remember the title, just the sense that if you ever want it, the longer version of this story is waiting there for you. For now, you can let the question stay open. What did Denisovan songs sound like, if they sang? What stories did they tell their children about the stars, or the wind, or the animals they hunted? Did they have names for us, the way we now have a name for them? We may never know, or one day some new discovery may give us the faintest outline of an answer. Either way, the fact that we can ask these questions at all is part of their legacy. They survived long enough and left enough of themselves behind that another branch of humans, us, can sit quietly in the dark and wonder about them, about the ways their lives and ours are entangled, about how many other lineages once walked beside us in the night and are now known only through the codes they left in our blood. As sleep comes closer, you can imagine all those branches, modern humans, Neanderthals, Denisovans, ghost ancestors in Africa and Asia, growing, meeting, fading, merging, not as a hierarchy, not as a ladder, but as a dense living thicket of kinship stretching back through time. Somewhere in that thicket there's a path that leads to you. For this moment, that's enough. You don't have to solve the mysteries or remember the dates. You can just lie there, breathing in and out, carrying the quiet knowledge that you are not alone, not separate, not new. You are the latest chapter in a very long story, written partly by hands we will never see, on landscapes we will never walk. The lost humans are not entirely lost. They are folded into the way your cells work, the way your body meets the world. They are with you in this small, ordinary act of lying still in the dark, feeling the pull of sleep. And as you drift, the story of all those ancestors, Denisovan and otherwise, keeps flowing quietly forward, through you, into tomorrow.